Hello.
So very good morning, sir.
Ah,
Sir, shall we start?
so
If you permit, sir.
is possible for us to start or we have some time?
Sir, if you want that to some time, you can take your time and then you can please communicate to us. Then we will start, sir.
Okay.
Good morning, sir.
Good morning, ma'am.
Good morning, everyone.
Good morning, ma'am.
Morning, madam.
Good morning, ma'am.
Good
Hey,
morning.
ma'am.
Good morning.
Good morning, ma'am.
Good morning.
Good morning, ma'am.
Good morning.
Good morning, ma'am.
Good morning, ma'am.
Good morning.
Good morning, ma'am.
Good morning.
Good morning.
So, friends, yesterday I enjoyed the all four sessions. Today, we're going to discuss the 4 different aspects with the 4 different experts. That is, first is the bio-nano-composite for the sustainable development. Another one is the molecular dynamics solutions for the chemistry teachers. Then next generation academia, integration of AI into teaching, learning, and research. And last one is the statistical analysis for the chemist. So today we're going to discuss these 4 different aspects with the help of 4 different experts.
Let me just add one more thing. These lectures, they are very important and it will be very, very beneficial. Those who are working in, doing research in nanotechnology, or suppose anybody is doing docking, so they will come to know what is simulations are. Maybe if it is possible for the resource person, he is going to demonstrate the things, how the free softwares are going to work for this molecular dynamics and the simulations and the docking and the other things. And you will get in third session, you will get an idea about the AI tools, how that can be used in teaching, learning, and research. That is again very, very important nowadays. And the last one is those for doing research in analytical chemistry, basically statistical analysis of chemistry. He's again a very good resource person. So all 4 lectures, I think today, these are very, very important.
Recording in progress.
Hello, Dr. Gade sir.
Yes.
Shall we start, sir?
Yeah, sure. Definitely. I was just waiting from your side. Sorry.
Yeah. So,
It's audible first.
yeah, sir. Very much clear, sir. Very much clear.
Okay, that's it. So,
Okay, so good morning,
thank
one
you,
and all.
ma'am, for giving at least opportunity to discuss
just-
whatever little bit which is work which has been done. The
Sir,
work
just
itself
wait
is
for
at
1
nano
minute.
level. So you
Gadesh
can
sir,
imagine the length scale, the work which has been done. I would like to share with you all. And
Dr. Gadesh sir, please wait for 1 minute only. We'll take first introduction and then we will start with the session. So now, first of all, may I request Dr. Baral sir to please introduce our guest in brief and then we will start with the session. Yes, Dr.
Okay.
Baral, please.
Yes, so good morning to the organizers and all my fellow classmates at this refresher course. And thank you, organizers, for giving me the opportunity to introduce today's eminent resource person, the first session of today. So Dr. Aniket Gade have a great fascination towards interdisciplinary sciences and does not remain confined to strict boundaries of any science disciplines right from his undergraduate days. He pursued his undergraduate education in microbiology, chemistry, and botany from Amravati University, followed by post-graduation in biotechnology from the same university. He has completed his PhD in 2012 and focused on microfabrication of silver nanoparticles. He then has a postdoctoral stint at Utah State University in US in 2013-14. So Dr. Gade served as assistant professor for 17 years at Amravati University till 2022 before moving to Institute of Chemical Technology, Mumbai as an associate professor, where he is currently working. He boasts a rich academic journey spanning over 2 decades, and his expertise lies in bioinformatics and nanobiotechnology, and have more than 100 publications and several patents. Dr. Gade's pioneering research focuses on microfabrication of nanoparticles and their applications in antimicrobial therapies. He is recognized globally and received prestigious awards and grants for his contributions, earning him the International Award from Biotechnology Advances, as well as inclusion in Stanford University list of top 2% most influential scientists. His recent accolades include the Colores-Beast Marie Curie Fellowship from 2023 to 25, highlighting his continued impact in the field. His work explores the potential of fungal mediated synthesis such as Fusarium and Phoma species in producing nanoparticles with antimicrobial properties, investigates the efficacy of these nanoparticles against multi-drug resistant bacteria, pathogenic fungi, and viruses. Dr. Gade's contribution to micro-nanotechnology and green synthesis methodologies underscore his commitment to sustainable and biocompatible nanoparticle production. His interdisciplinary approach bridges biology, nanotechnology, and material science, which paves the way for novel therapeutic inventions and solutions to the environmental issues. So today we will have the opportunity to see some of these in his lecture entitled Bio-nanocomposites for Sustainable Development. So welcome Dr. Aniket Gade sir, and please start today's session. Thank you.
Thank you, Dr. Abhishek, for such a wonderful introduction. As such, with respect to all those credentials which we have, like publication and like fellowships and patents, in course of a time, probably each one of you, those who are as a participant, is going to have all those credentials. So it's mainly with respect to what little bit I could understand about a topic which I'd like to share with you all. Might be useful. I don't know how far it will be useful with respect to the area which you carry out the research, but somewhere or the other, it will help you out. Because that's what many a times, if you have the experience of others, probably it does help us to progress, make a progress in our own field. So with respect to the today's, it's not a lecture, I would say. I'll make it clear. So it's not mandatory that you need to complete a presentation and then you can ask a question. Whenever you have a question, you can stop, you can directly ask a question. So it's going to be like just kind of an interaction, I would say rather. But to progress the interaction, I do need some kind of a point. So I have prepared a PowerPoint presentation for that. Not necessary. Our aim is not to complete the slide. The aim is to understand with respect to a topic. So today we are just going to talk with respect to bio-nanocomposites. So I'll just would like to share a presentation with you all. Let me know whether the slides are visible to you all or not. So
Yes, sir, it is visible.
slides are visible?
Yes, sir, yes.
Okay, I'm just going to the starting slide rather than just starting in between, and we'll make it into a presentation mode. Hopefully the slides are visible. As we are going to discuss today is bio nanocomposites. This is one complex term, and then it's going to be how far it's going to be useful for sustainable development. There are several applications which have been there for bio nanocomposite, but I'd like to just focus on the kind of application on which I have worked. You have your own choice to work with those applications. So just to begin with our journey, these are the kind of topics on which we are going to focus. So we are going to see first what is bio nanocomposites. So once you understand bio nanocomposite, it consists of two major components, a biological constituent and a nanomaterial, or we can say nanoparticles, which is a part of a nanomaterial. So these two things, if you combine, it becomes a composite. So we are going to see nanoparticles, what are nanoparticles, and then we'll have idea about some of the biological constitutions which have been used. And then we'll see what do you mean by sustainable development. This is initial part with the introductory to understand with those terms. Once these have been understood, then how we can go on applying this kind of a work for in a research. So this is how the flow is going to be there. I just wanted to have a basic flow, but while going through with any one of the slide, it's possible you can stop or you can interrupt any time, and if you have any query, we can discuss. So just to begin with, as I mentioned that bio nanocomposites, this is a long term, and this term can be broken down into three parts like bio, nano, and composites. So bio simply means it has got a biological origin. Nano is going to have a nanomaterial or nanoparticles, and composite is a combination of two things. So we are going to have a biological constituent to which we are going to add a nanomaterials or nanoparticles to have such kind of a combination or composite with some unique properties. We are going to make it. This is what we are going to call it as bio nanocomposites. So now bio nanocomposites has got two important components. One is biological constituent, another one is nanomaterial or nanoparticle. So first we'll see what is nanomaterial or nanometer scale. So we all know with respect to nano prefix, nano itself has got a Greek origin where nano means dwarf. And just to get an idea how small a nanometer is going to be is like 1 billionth part of a meter is going to be a nanometer. It's quite easy to tell on a length scale that 1 billionth part of a meter will be equal to a nanometer. But sometimes it becomes difficult to visualize or assume how actually small this 1 nanometer is going to be. That we'll see. So first with the definition is quite easy. 1 billionth part of a nanometer or 1 millionth part of a millimeter could be a nanometer. And we know that we have atoms which has got the size in Armstrong. So 10 Armstrong is equivalent with 1 nanometer. So several atoms which are coming together to form a cluster which is going to be in nanometer scale. Now it says that, are you going to call any material as a nanomaterial? So a material having at least one dimension. We know there are 3 dimensions which exist for a material. X, Y, Z axis, we know it. So if any one of the axes, if you have in a nanoscale is 1 to 100 nanometer is a nanoscale. So if you have at least one dimension in a nanoscale, we'll call it as a nanomaterial. Nanoparticle is a type of a nanomaterial which has got all its dimension in a nanoscale. So that's how we are going to see with the nanoparticle. Now there are in a biological system materials available with having dimensions in a nanoscale. So those dimensions in a nanoscale, everybody knows that we have got DNA as a genetic material, it has got its dimension in a nanoscale. The thickness of a DNA which is there is 2 nanometers. So that's the one of the dimension in nanoscale. So you can call DNA as a nanomaterial as well. Even most of the biomolecule has got their dimensions in a nanoscale. So if I just want to give an analogy, trying to understand how small a nanoparticle is. This is a simple analogy which is being given on the slide. If I have a carbon dot, or I would say a quantum dot, which has been there. Now this is quantum dot, which has been made by using the different ions which have been there, a cluster of ions which makes one particle which has been there, which is quantum dot. Now, this quantum dot is equivalent to a size of a football, because right now we are having a FIFA World Cup, which is in progress. So it's better to give analogy in terms of a football, which goes on with the topic. So quantum dot, which correlates with respect to a football, if it is equivalent to football, then actual football will be equivalent to a size of a mother earth. So this is what we are going to assume with respect to the nanometer scale. There are a few more examples. If you want to understand, the thickness of a paper sheet is almost 100,000 nanometer thick. We already talked about, like, strand of a human DNA is 2 nanometer in diameter. A human hair is approximate 80,000 nanometer thick. So that's about trying to understand and visualize how small a nanometer is there. So the question comes like, when we have like bada hai to behtar hai, then why you want to go at such a small size material which has been there, which is not even visible with the naked eyes. So what happens if you have the same material, if you go at nanoscale? One of the important change which happens is due to a surface area to volume ratio. As you go on decreasing the size of a matter, there's going to be a larger surface area to volume ratio. And this parameter is largely responsible for affecting the properties of matter. So it changes the reactivity and it start behaving differently. Though the origin is same, but due to size, it behaves differently, so it has got a different properties. So at nanoscale, what happens, the material is going to have larger surface area to volume ratio. There's going to be a size confinement effect, which has been demonstrated, large number of surface atoms and reduced energy, which will be there, increased surface tension, decreased gravitational force. So as a packet, as a whole, due to all this, what happens in a simple language, the properties of a matter at the nanoscale changes as compared with the bulk. So if the property changes, which all properties, the properties which are going to change is going to be optical properties. Say, well-known example is with respect to gold. Everybody knows how does the bulk gold look like, and gold nanoparticles, they have got different color. So that's the change in optical property. It can be easily been demonstrated for gold, for silver, even for copper, which has been there. Sometimes it helps in changing the electrical property with respect to conductivity, physical properties like hardness and melting point. You have like changing a chemical property with respect to reactivity, change in the reaction rate, or change even in the magnetic property. So change in the property is what we mentioned. It can be any one of these properties which would be changing. You need to demonstrate which property has been changed at a nanoscale level. So at nanoscale, materials are going to have strikingly different properties as compared with respect to its bulk counterpart. So the example which we mentioned previously is the same one. It was with the gold. Now you could see here on the slide itself, there are different size of a nanoparticle will give you different color, which has been seen. So when you have same shape, but the size is different, it behaves differently. Similarly, if I have got different shapes at nanoscale even, it will behave differently or will give you different colors. That means different with respect to the optical properties which is being seen. So as such, what we could thought, there is a lot of literature which has been available. The change in the properties is due to a change in the size and shape at nanoscale. What we understand while working is not only size and shape, but it's going to be the concentration, which also matter with respect to changing the structure and properties of a nanoscale matter. So it's the size, shape, and concentration. This together is responsible for changing the properties at nanoscale. So once it has been clear why you want or why you are interested in getting a nanomaterials or nanoparticles synthesized, so you should understand the methods which are being used to synthesize nanomaterials. So there are two broad classifications. The first classification has got two approaches. One is top-down approach, another is bottom-up approach. The meaning lies in the name itself. When you say top-down, that means you start with the bulk material. You cut this bulk material, carved it, grind it, and bring it to a nanoscale. So we call it as top-down approach. Another approach is bottom up, where you start with the precursor, which is smaller than nanoscale. So generally we go on with the precursor salt. When adding to a water, it dissociates into ions, and ions has got the size in Armstrong level. So these ions which will be coming together to form a nanoparticle, which will have a size into a nanoscale, that is 1 to 100 nanometer. So this we call it as bottom-up approach. So this is one classification which has been there. Based upon these approaches, there are different methods which have been available. So for example, top-down to top-down approach, the methods like mechanical processes, thermal process, sputtering is there, chemical etching is there. Then you have a bottom-up approach, the methods like vapor deposition, salt shell, wet electrochemical method, bioreduction comes under this. So the methods can be grouped into top-down and bottom-up approach. This is one classification for the methods which are being used to synthesize nanoparticles. Second classification includes which approach you are going to use, whether it's a physical approach, chemical approach, biological approach. Apart from this, now there's one more classification which we call it as a hybrid approach, where you can mix physical and chemical or chemical and biological. That combination can be mixed to go on with the hybrid, which is not been listed here. I just wanted to show you there are three basic methods to synthesize a nanoparticle. Physical method, in case of a physical method, all those physical methods have been classified into two types, mechanical one, another one is vapor-based. In case of a mechanical method, the example is milling, mechanical milling, which has been there, ball milling, where with the help of heavy metal balls, we just go on grinding and making particles in the nanoscale. Vapor method, a physical vapor deposition, which is there, which includes laser ablation, laser pyrolysis, or sputtering. Then you have chemical approach, salt shell, chemical vapor deposition, colloidal methods, spray pyrolysis. And I'll be interested mainly because I'm working on biological approach, where we make use of a biological system or a part of a biological system to synthesize a nanoparticle. And it's simple, like each bacterial or fungal cell itself is biochemical factor, so which keeps on carrying out several reactions. So I want to use a biological system to carry out a synthesis of a nanoparticle. So that's what we call it as a biological approach to synthesize a nanoparticle. Now, in case of a biological approach to synthesize a nanoparticle, either you can synthesize a nanoparticle inside the cell, we call it intracellular, or outside the cell, we call it as extracellular. Now, the approach which has been there, because I can grow bacteria or a fungi at normal temperature or room temperature is possible, and at ambient temperature and pressure, it's possible for me to culture this organism. So these methods are going to be quite eco-friendly, and moreover, you can always carry out the biological system can be grown at any particular bacteria can be grown up on a large scale. So the scale up of a system should be even possible to grow a particular bacteria or a fungi on a large scale. So these methods which are either can synthesize inside the bacteria or a fungi or outside. If you synthesize the nanoparticle inside the cell, naturally you need to sacrifice a cell to collect the nanoparticle. It's better if it has been synthesized outside the cell, so you don't need to sacrifice a cell to collect your nanoparticle. So extracellular is a better approach as compared to intracellular. Now there are reports we can make use of any biological system like bacteria, simplest of a biological system, fungus, plant, algae, animal. You name a biological system that can be already been reported would have been there to synthesize the nanoparticle. Preferably, why you go for synthesizing with why you want to go for synthesizing the nanoparticles which have been there is because this if you make use of a biological system to synthesize the nanoparticle, it's possible to have a easy scale up because I can grow a bacteria, fungi on a bulk scale. Since it has got a biological origin, these are going to be biocompatible, where biomolecules generally act as a capping agent and as well as a reducing agent. It's possible to interconvert a polite to a powder form and powder to polite. Now, this is mainly been required when you want to go for characterization of a nanoparticles by different methods. So that is there. Even in case of a downstream processing, it's going to be quite easy if the extracellular synthesis is being carried out by using a biological system. So considering all these advantages, it says that it's better to go for synthesizing nanoparticles by using a biological system. Now, you all are chemists, you know that synthesizing a nanoparticle generally require three things. One is solvent, then you require reducing agent, and then you require stabilizing agent. So with the help of these three, it's possible for you to carry out a synthesis of nanoparticle. And as per Environmental Protection Agency of USA, they have given some norms if you want to carry out any nanoparticles, because nowadays not many nanoparticles are coming into a consumer products. There are sunscreen lotion, there are clothes, there are other products which have been there which we use has got an involvement of a nanoparticle, but it has told that the nanoparticles can be used, but this nanoparticle should be synthesized by a green approach. So in case of a biological synthesis, naturally, if you want to have a green approach, the three things which needs to be concentrated is solvent choice, environmentally benign reducing agent, and non-toxic stabilizing agent. So these three things which have been there, in case of a biogenic synthesis of nanoparticle, generally we use a solvent which is a water, and water is a universal solvent. So in case of reducing agents, it's generally you have biomolecules serving as a reducing agents. So these are benign reducing agent as such, which can be either protein, sugars, or phenolic compounds, can carry out, can act as a reducing agent. And the last one is stabilizing agent. So stabilization of an agent even is being done by the biomolecules. So biomolecules like protein acts as a stabilizing agent. So it fulfills all the three conditions which have been given by Environmental Protection Agency of USA. So the synthesis of nanoparticle by using a biogenic approach is going to be a green process or green approach. So that was with the first part with nanoparticle to give you some introduction. Again, we are coming to the same slide. From here, the second part is with respect to biological constituent. So why we should be using a biological constituent while making a composite? One of them is nanoparticle, where nanoparticle can be synthesized by using green approach. Now we have a biological constituent. So biological constituent because it's renewable. You can produce biological constituent in a bulk quantity as and when required. It's going to be abundant in nature, renewable, low cost. It can have effect on solubility. It's non-thermoplastic. So on heating, it's not going to change its shape. Biodegradable, which has been there, and biocompatible. So considering all these properties, it says that it's advisable to go for using a biological constituent for forming of composites, where the one component is biological component constituent, another one is with the nanomaterial. So some of the natural biopolymers which have been exploited for the synthesis of bio-nanocomposites are like chitosan, cellulose, starch, zinc, collagen, casein, gelatin. So these are few polymers, biopolymers, which have been exploited by each one of them. Now, I'm going to tell you like what we have done in our study. We have made use of a casein for forming the composites. And so when we'll discuss our work, probably I'll be able to discuss why we choose to use casein. There are some synthetic biopolymers as well, which can be used, like polyglycolic acid, PGA, which has been there, polyhydroxyalkanone, polycaprolactone, PCl, polyvinyl alcohol, PVA, polylactic glycolic acid, PLGA, and polylactic acid, PLA. Even these are which can be used for the preparation of bio-nano composites as well. So if you could say a biological cell which has been made of different biomolecules, there are 4 major types of biomolecules which are present in any living system. And that's going to be like carbohydrate, protein, lipids, and nucleic acid. So all of them can be utilized for preparation of which each one of them can serve as a biological constituent in preparation of bio-nanocomposites or biocomposites which are being made. Lecithin we use, which is a part taken from a membrane, hydroxyapatite, which has been taken from bone cell, which has got the calcium, then cellulose and starch. These are well-known polymers which we know. So you can get it from cotton, wood, soy, hemp, flax, jute, even from banana peel is possible to get it. Then you have chitin. Chitin has been taken from the exoskeleton of most of the crustaceans like crab, which has been there. This chitin is present, we can get. Then protein is one of the common biopolymer which has been used, which can obtain from soy, wheat, gluten, which we have already seen the examples earlier. Then you have polyhydroxyalkanones, which have been there, and then the nucleic acid. So hopefully the second component is clear. First, we discuss with nano material or nanoparticle. Then we have like biological constituent. Now we'll try to understand sustainable development, and then we'll correlate the formation of bio-nanocomposites with respect to a sustainable development. So as per the United Nations, the definition for sustainable development stands for which meets the needs of the present without sacrificing the ability of future generations to meet their own needs. So This is what we call it as sustainable development. And based upon this definition of a sustainable development, it has got three important components. Those are like environment, economic, and social. So integration of these three components, which is a key in case of a sustainable development. So any sustainable development approach will try to meet these three issues: environment, economic, and social. So then we can, if all the three areas have been, or three issues have been overtaken, then we can say that our approach is sustainable. Now, United Nations has given sustainable development goals. There are 18 such sustainable development goals which have been enlisted by United Nations. If you could see, look at all those 18, this can be grouped further into 5 P's, starting with the people, production, then planet, which has been there, peace, and so all those 18 goals as such can be classified, and naturally the effect on trying to achieve one goal will affect the another goal as well. So it says that they are interlinked, all those 18 sustainable development goals which have been there. So just coming back to our basic topic with bio-nanocomposites, it has got applications in several areas which have been there. So till this, hopefully the things are clear. This was the introductory topic. We just wanted to introduce with all those terms, because not necessary that everybody will be working in the area of bio-nanocomposites or composites. So we wanted to start with the scratch. Now we'll go on with respect to the kind of work which is been done by us. It's quite easy to tell in theory that needs to carry out a synthesis of nanoparticles by using biological system. But when you actually carry out synthesis of nanoparticles using a biological system in a lab, there are going to be several obstacles which will be there. You need to overcome those obstacles to get the nanoparticle. So since my supervisor, that is Professor M.K. Rai, he happened to be a mycologist, so and he knows a fungal system very well, because with respect to his experience. So we decided initially to concentrate on fungus for synthesizing nanoparticles. So we exploited several fungal species like Fusarium, Phoma, Alternaria, Aspergillus, Pestulutia, higher fungi which have been there, or we call it as a mushroom, even were exploited for synthesizing the nanoparticle. Now, this was our first paper which was being published in 2008 when we started our work on nanoparticle synthesis by using a fungi. So here we isolated a fungus from the ginger, which was infected with fungi. We could isolate those pathogenic fungus, identified it, and then after culturing this fungus in a lab, we synthesized the silver nanoparticle by using this one and demonstrated its antibacterial activity. So just to initiate our journey with fungus, it happened that when we started our work in the Amravati University Biotechnology Department. There were many facilities with the characterization of a nanoparticle was not available. So when the things are not available with you, but you have a concept, we wrote a joint project, and this was the joint project with India and Brazilian counterpart in the Campinas University. So Indo-Brazil project, which was being sanctioned by DST, and under this project, we were able to synthesize the nanoparticle, and the characterization facility was available in the University of Campinas, Brazil. So where the characterization was being carried. Now, when we just wanted to see that, because silver ions has got antimicrobial activity, nanoparticle has got antimicrobial. If both are going to have the same antimicrobial activity, there is no logic in synthesizing the silver nanoparticle. So we wanted to see what is the kind of a difference which is being there. Now, in case of a silver ions, generally silver ions, if it comes in contact with chloride ions, it tends to form a precipitate of silver chloride. Whereas it doesn't happen with nanoparticles, which is a cluster of ions, and it will release ions at a rapid rate and at a particular position, which doesn't happen with randomly suspended ions. So we could see how does it kills a bacteria. It says that the nanoparticles will get attached on the surface of a bacterial cell, and then from that surface attachment, it will start leaching out ion inside the cell, which will bring about the rupture of a membrane and in the end, the lysis of a cell. So that could be analyzed and reported in our publication, which has been there. So we started with the silver, which was easy to synthesize. Nowadays, if you just give a keyword with the silver nanoparticle, you get lot many publications where the nanoparticles have been synthesized by all different fungi is possible to do. So we don't want it to restrict ourselves with the silver. So we could synthesize other types of nanoparticles as well. Iron oxide nanoparticles we could synthesize. Now, this is a kind of a study which was been carried out during my Raman Fellowship, which was given by UGC. Had the opportunity to work in the University of Utah Biological Department, which was been there. My mentor, Professor Ann Anderson, who was been there. She was been working on environmental friendly bacteria, Pseudomonas chlorophyllus O6 strain. She was working on that. So she wanted to check if this bacteria, which is environmental friendly bacteria, if the silver nanoparticles comes into environment, what will be the effect on this bacteria? So that kind of a study which we carried out, we happened to work most of the analysis which was being done with the change in the morphology of a bacteria by atomic force microscopy. So bio-FM system which was being used to analyze those bacterial cells. Now, one of the constraint is biosynthesis is using a biomass for synthesizing of nanoparticle. But the constraint is, can this biomass is being used continuously? So we wanted to check. So this was the concept or kind of a question came into the student who was there, master student. And he said that, is it possible to use repeated using of a biomass? So we went for encapsulation of biomass in a sodium alginate bits and wanted to check. So it was possible for us to carry out 10 batches of silver nanoparticle synthesis. Of course, each batch, there was an effect with respect to the zeta potential, that is a charge, or even the size has got the effect. So there were change or there were effect on the size and potential of a particle which was been there, but it was possible to synthesize 10 batches of a silver nanoparticle with the immobilized biomass which was beneath. Then we also synthesized copper nanoparticle, which has copper nanoparticles found to have antimicrobial activity, as well as copper serves as a source of a nitrogen, the source of a copper to the plant, which is one of the micronutrient for the plant. So copper nanoparticles which were there. Then this is the one, once you synthesize a particle and you know that this particle are effective, next step is to go on preparing some kind of a formulation. So we prepared the formulation of silver nanogel, and this silver nanogel was carried out to check for wound healing activity. So this was the publication, but this was the work based upon a project which we received from Rajiv Gandhi Science and Technology Commission. And we happened to even patent this particular silver nanogel as a product. So, which has got wound healing activity, which was being demonstrated by this particular silver nanogel. So, this was the composite formation. Silver nanoparticles were entrapped in polyvinyl alcohol film matrix. Still, it could demonstrate its antimicrobial activity and so on. So, in short, I'm not going to tell you everything, but just to give you a bottom line, we have got several publications where we could exploit using different fungus for synthesizing nanoparticles. Now here, mostly we are concentrating on fungus, but we have work even with the plant extract, even bacterial extract for synthesizing, even algae were exploited for synthesizing nanoparticle. But for us, we found to have a fungal as a better system for synthesizing of nanoparticle. So that was the first part, synthesizing the nanoparticles by using a biological approach. Now, if I'm going to use nanoparticles anywhere, naturally the nanoparticles will be attacked by different organisms and then start leaching out ions and will be it will be converted into all the ions. And the most of the activities are at nanoscale if it is remain at this particular. So can we allow to release the ions as and when it is been required? Nanoparticles are there. Nanoparticles are cluster of ions. When they are supposed to release this ion, is it possible to control that? So that was the concept. And for that, you need some sort of a carrier which will hold all these nanoparticles and will start leaching out ion as and when we need it. So for that, we are supposed to provide some or the other type of a stimulants. So that the carriers which have been used for control or sustained release, it can be a drugs, it can be a nanoparticles which will serve as fertilizers, pesticides, or even enzymes or other nutrients. So carrier matrix should be there, and then you should have some sort of a response which will trigger its release. The different kind of a response which can be utilized in releasing of ions could be temperature. So by changing the temperature, it should be possible to release the ions. pH responsive, so like changing the pH, which is possible for us to change the pH, like we can make a pH acidic, we can make pH much more basic. So we know how to do that to change the pH. We can change the magnetic response, we can change the light response or photoperiod, moisture response, enzyme response, or sometimes the secretions. So all these can serve as a kind of a response if they want to release the nanoparticles. So considering this, based upon this concept, we happen to write a grant to a European Union, and this was the grant which we have written. The title of a grant was Calcium Carbonate Encapsulated Casein Nanocarriers, which were having nanonutrients, which serves as a source of a plant nutrients, which was being. So it was a novel nanocomposite fertilizer. This was the title which was being there. Actually, this project was being based upon control release of fertilizer. Nowadays, we do make use of chemical fertilizers. If I spray a chemical fertilizers naturally in a farm, those fertilizers will be taken up by plant, rest of them will get washed away. So it says that almost 60% to 80% fertilizers which have been applied to a soils go as a waste, which cannot be taken up by plant. And plant cannot be taken, is not going to take all those fertilizers at a time. It needs, with respect to its growth and development, the steady supply of this fertilizer. So the concept was like we'll have fertilizers which have been added, but those fertilizers which will be released into the soil in a controlled manner, in a slow manner, so that even plant could maximize its uptake and we can minimize the loss of fertilizer which happens with the conventional fertilizers. So that was the concept on which we got this particular grant called as control release fertilizers, which we got it from European Union. So in this particular Grant, as it has been mentioned, like we just wanted to have a nanocomposite which is going to serve as a nutrient and it will release the nutrient in a controlled manner. So we know that the plant has got requirement in the form of macro and micronutrients. And we could synthesize nanoparticles. The nanoparticles, like plant has got the requirement for copper, will provide copper nanoparticles. Plant has got the requirement in terms of zinc. We can provide zinc oxide nanoparticle. Plant has got the requirement in terms of Mg, Mn. We can have their nanoparticles. Plant has got the requirement in terms of iron. We can prepare the iron oxide nanoparticles and can provide as a nutrient. So to supply a nutrients, probably this slide, if you could understand, I would say, we'll understand the whole scenario in a project, what we were able to achieve. Like we were concerned with respect to synthesizing the nanoparticles, which could serve as a source of nutrient to a plant. These nanoparticles were involved with forming a nanocomposite by using a biological constituent. The biological constituent which we have used in our study was a milk protein named casein. So once you form this biocomposite, again this biocomposite was given additional protection in the form of a layer called as calcium carbonate. So calcium carbonate along with protein, it could form a microsphere, and inside which you have your nanoparticles and even the proteins which has been present. So when this complex is present, as such, the outermost layer is going to be of calcium carbonate. Now, calcium carbonate is responsive against the pH change. At acidic pH, this calcium carbonate complex is going to get collapsed. So it will tend to release the composite which has been present. Now, in a composite, we have got protein, which is milk protein like casein. So proteases will degrade this protein and will release the ions. And once it releases the ions which are from the nanoparticle, that will be taken as a source of a nutrient. So this was the whole thing which we wanted to carry out in this particular proposal. So the stimulus we can receive from plant is in terms of the root exudates which have been shown by the plant, as well as change in the pH which will be there, or even it's going to be with respect to secretion of proteases. These are the kind of response which we use it from the plant to work with respect to our nutrient. As I mentioned, even for any particular thing, you should have some logical justification why those things were chosen in a project. So we happen to choose to work on copper, zinc, Fe, and Mg. It's mainly because out of these four, the copper, zinc, and iron, which serves as a micronutrients to a plant, and Mg serves as a macronutrient to a plant. So we develop a method to synthesize the nanoparticles of this all the four, and then their complex was being prepared. And this complex, prepared complex which was being there is the one which was being used to demonstrate the effect. So say for example, we have screened this many fungus to synthesize all these nanoparticles which was been there. So where we could found that particular fungus with zinc oxide, Mg oxide, copper, and Fe nanoparticles, after screening those fungus, we could optimize each one of them. And we went for synthesizing all these nanoparticles by using those fungal extract. So fungal extracts, once we could synthesize, naturally all those nanoparticles were characterized by using different methods. Those nanoparticles which were available. But now, because nanoparticles, if I'm going to give it in the form of a nutrients, will start releasing or leaching out the ions, and it's no more is going to remain in a nanoparticulate form. So I want these nanoparticles to be protected. They should not leach out iron. They should leach out the ions only when it has been required. So I'm going to form a complex with, or I'm going to form a composite. To form a composite, we chose basically a biological constituent like milk protein, or we call it as casein. So this milk protein, which is biocompatible and non-toxic, it's available. It is basically, casein is amphiphilic in nature, which helps a lot. Inexpensive, readily available, less immunogenic, and it has got several function group, because we know that casein has got ability to bind with the calcium, and that's the another reason why we particularly chose to work with the casein. So considering all these reasons, we chose to have a composite of nanoparticles along with casein. Now, you could understand this schematic representation gives you an idea how did we proceed for the preparation of a composites. So, casein micelle, which has been there, at particular pH, which has been there, is going to be in the form of it's in the form of a small micelles, which has been. So, if I'll increase the pH, make it much more alkaline, now this micelles gets open up. So at alkaline pH is what we are going to add a nanoparticle. A nanoparticle has got the affinity for the proteins which are being there. So they'll interact with the protein and again will reduce the pH, make it acidic, so that again it will form the micelles. Now the micelles are with the nanoparticles which are being there. Now this is the schematic way which was been there, but you need to carry out the experiment to understand. So we could prepare a complex with casein and copper oxide nanoparticles. We could optimize the concentration which has been required, and so on. So each one of the complex which was been prepared to make you understand. Now these were the kind of a complex with the composite which we got along with the nanoparticles which were already been entrapped inside this particular composite. So these composites which we prepared, so we prepared for copper, we prepared for iron, we prepared for Mg, we prepared for zinc. So all the nanoparticles which were entrapped into a protein casing, so we call it as the short form CUMC stand for copper nanoparticles with micelles of a casing which have been there. That was the complex which has been there. Naturally, you need to go on characterizing to confirm that actually the complex has been formed. Now, again, this particular complex is not going to be that stable, because if I'll add the bio-nanocomposite to a soil extract, immediately it will be degraded by all the microbes and other things which have been there, because each one of them will secrete the protease and they'll carry out a degradation of casein and release of ions. So I need to have one more level of a protection to this complex, and that has been given by calcium carbonate. So why calcium carbonate? Because calcium carbonate, which are biocompatible, safe, calcium carbonate are pH sensitive with respect to their dissolution. As I mentioned earlier, this was change in pH was one of the kind of a response which we have decided to have for releasing of nanoparticles by the calcium carbonate microsphere. Then these microspheres are going to be porous. So again, porosity and surface area is quite easy to fabricate, quite easy to say with that is easy to fabricate, but it do take some time and optimization to actually carry out the fabrication of calcium carbonate microsphere. It's going to be eco-friendly, it's going to be cost-effective, compatible, and it's possible to functionalize. So considering all this aspect, we decided to choose calcium carbonate to have as a additional protective layer or this additional protective layer with respect to our composite which was being prepared. So now this is a paper which we have published in 2025. Now it's been present. So here we could demonstrate that initially calcium carbonate can form the nanoparticles, and these nanoparticles are with respect to the calcite isoform of calcium carbonate which has been there. These nanoparticles can be assembled to form a microsphere that we could demonstrate, and to forming a microsphere, the protein is going to help, and the casein protein does help in forming a microsphere. Now, this was the work which was casein-assisted biomineralization of a calcium carbonate microspheres which was been there. And it has got enhanced properties which was there. It's mainly with respect to the water retention ability. If you could carry out water absorption and desorption analysis, the kinetics is somewhat similar, but only the difference is that the rate at which the desorption happens is almost at a half of the rate to in presence of a casein as compared to without casein. So that was the difference which we could see. So with respect to water absorption desorption study, we can say that there is a better water retention ability which exists with respect to the calcium carbonate microspheres which have been formed with the help of a casein. Now, here you could see how actually the microspheres could be formed. So here both the complex in presence of a casein and in absence of a caseins have been shown on the slide. You could see that the one which is without a casein, which has been there, it shows like having small particles. It's not perfect sphere. Whereas when you add a casein, it shows a perfect sphere and smooth surface could be quite easily been visible. And these microspheres, they are going to be of similar size, having a diameter of 2.65 micron. So they are a bit large in size, which have been there. So it's possible to encapsulate a casein along with the nanoparticles, which can act as nutrients in this particular complex. So this way, we could, as such, synthesize the microspheres having encapsulated nanoparticles in it. So we could have the microspheres of calcium carbonate encapsulating CUMC, which stands for copper nanoparticles along with casein. Similarly, zinc oxide nanoparticles with casein, iron oxide nanoparticles with casein, Mg oxide nanoparticles with casein. So all those complexes, and you could see there's a difference with respect to each kind of a microsphere which has been formed. We didn't want to go at that minute level because we wanted these nutrients to be, or the composite to be encapsulated in a calcium carbonate. And after encapsulating, they should start releasing out ions in a controlled manner. So that was our aim to have. So we could prepare. Now it was, we could get microspheres of iron. They are of larger size. The reason was that we could not provide same kind of a stirring speed because these iron oxide nanoparticles has got a magnetic activity. So you cannot make use of a magnetic stirrer, which I could use it for copper, Mg, and a zinc oxide nanoparticle. So for iron, I just have to rely on the rotor which was been there. So that's not going to be sufficient. That speed is not going to be at comparable with the speed which has been achieved by magnetic stirrer. So due to the less speed, the size of a microsphere is a bit larger as compared to the microspheres which we received from other kind of a complexes. So size of a Fe casein microsphere is almost 11 micron, whereas of the copper it was 1.52, it was Mg casein it was 4.67, and you have 1 micron with zinc. So that was a variable size which was been achieved from each one of them. Now, even once you receive those microspheres, you need to characterize actually, does it have got all the ingredients which you are planning? So we could characterize those microspheres. You could see that there is a pore shape kind of a structure which has been seen or has been visible. So you do have composites, and above a composite, there's going to be a layer of a calcium carbonate. And with respect to EDAX analysis, we could find out the elemental composition of a copper along with the peak for a protein which have been there, protein which has been made of carbon, hydrogen, oxygen, nitrogens which have been there. Generally, the protein serve as a source of a nitrogen, to a plant as well. So the further characterization which was being carried out to get it confirmed that it's actually your complex is been formed, the one which you wanted to have. So you can go for surface analysis, like nitrogen adsorption, co-analysis of those microspheres which you have synthesized. So it could say that the porosity was around like 5 nanometer was there. So it was mesoporous structures which were been obtained. Naturally, through this mesoporous structure, the enzymes could enter and can carry out a degradation of the proteins which are entrapped into the calcium carbonate matrix. So apart from this analysis, we could compare the water retention ability. Water retention ability, which was being done initially. So it says that zinc has got the highest water retention ability as compared to Mg, copper, and iron complex. So we could, but all those complexes has got better water retention ability in the presence of a nanoparticle as compared to when the nanoparticles are not present in the complex. So we could carry out this analysis and could confirm that a zinc complex has got a better water retention ability as compared to other complexes. So this was the first part which was been there, like we wanted to make up a complex. Naturally, we started with the concept, like we wanted to design a nanocomposite which should serve as a control release fertilizers. So on that basis, on that concept, we initially prepared a composite. Now composite has been prepared and is ready. Now you need to carry out a plant experiment to verify does it actually behaves the same manner the way you thought. So we carry out the plant experiment at a control conditions. So control conditions, when we carry out this particular experiment, and we carry out all these experiments against the maize plant, we grow all these maize plants in a control condition for 4 weeks. Naturally, you need to have several steps, and the experiments was being carried out in a sand. The logic was soil is a much more complex system. It do have a presence of some residual nutrients. And if you want to avoid the interference of these residual nutrients, it's better to work with a sand, so that whatever thing which is been added to a sand is only additional additions which have been there. Due to that, the effect would have been obtained. So naturally, you need to have all sorts of a control when you carry out experiments with respect to the plant. You need to have a positive control. You need to have a negative control. You need to compare with those. So we carried out all those plant study on a maize plant. After 4 weeks, we harvested those maize plant and then we compare. So when we compare, one of the component which we understand was comparing the chlorophyll content of a plant. What happens with a control? The control was basically we provide a media like Hoagland solution was the one which was being provided to a plant. That was for a control plant, whereas Hoagland solution minus the zinc component, where we have used our zinc complex, was provided. Similarly, we substitute for copper, Mg, and Fe components. So when we provided, it was been found that the chlorophyll content is been enhanced in the plant. So plant demonstrated higher chlorophyll content with the treatment of 4 weeks. Now, this was the experiments which were carried out in a control condition. Naturally, our second step is going to be, after carrying out the experiments on a control condition, would like to move on with the experimentation in a field. But we cannot directly move on with the field because we wanted to get it confirmed whether our complex which have been formed have been working fine or not. So we have got the comparison of all different things. Like we compare the shoot length, root length, which has been there, or leaves, stem, roots, which have been there, dry mass, their fresh weight, and all those comparisons, all those data which has been carried out. And after carrying out all those kinds of analysis, at least we are quite assured that the complex which we have prepared, the complex which have been prepared along with the nanoparticle. So we could say after carrying out this study, nanoparticle could serve as a source of a nutrient to a plant. One. Second thing, nanoparticles with the composite and having calcium carbonate does help with respect to slow release. Now, there is still, this is study which has been there because we just had 2 years of a time to work on all those things. And generally, the plant experiment for 4 weeks takes a lot of time. So still there is some work which needs to be carried out to validate these things. In vitro would like to go for carrying out the kinetics, which will demonstrate the control release of ions, which will happen from the complex which we have prepared. Now, this kinetics and control release experiments, the preliminary work which we have done, like still we'd like to go and get it confirmed with some more work and trying to understand what how does it behave in vitro. And naturally, once we are satisfied with respect to the kind of a response or kind of a data which we are getting with respect to in vitro study, then we'll move on with respect to the next level, and the next level is going to be applying these complexes in the field. So that is how the things will move on. But this project, which we work on, I've told you, at present, there is a greater demand with respect to the composites, which could demonstrate a controlled release of bioactive compounds, where the bioactive compounds could be your nanoparticles, it could be drugs, it could be any other molecules which have been important with respect to therapeutic point of view, there can be done. So as such, biocomposites with respect to a course of a time, at present at least it's the need of art, and it says that it has got the application, wide application into a different field. But you need to decide when you want to make any particular composite, which all the components you are going to select, and what is the logic why you want to select them to go for your study. So with this study, at least as I mentioned, we still have some of the work which we are deciding to carry out, and there's still some more work which will be done. It will be with respect to the kinetics or the study of nanoparticle release from this particular bio nanocomposites. So I say that bio nanocomposite because we use biological constituent, which is in terms of milk protein or casein, nanoparticles as a source of nutrients, and making their composite, which was been there. We use additional layer of a protection that was made by calcium carbonate. So that was the complex which was been formed, and we could make complex form, and we could demonstrate with respect to its application as well with respect to the plants and on the field. But it's going to take some more time. Like these are the study. Now we have filed international patent with respect to the synthesis of a nanocomposites. Still with the application, because it has got wide application. We do mention about applications with respect to the agriculture or as a fertilizers, as the application which has been mentioned. In case if it is going to be a control release, naturally is going to require less dosage of a fertilizations needs to be given to a plant during its complete life cycle, and the number of dosage of fertilization even will reduce. So that's the aim which has been there. But starting with the concept of nanoparticles which we are working, then we decided to move on with the building of a composite, that is bio-nanocomposites, and then bio-nanocomposites with respect to for a particular application. That's how the story moves ahead. Now, these are naturally, whenever you work anywhere, you require a team to carry out your work. So this was the team which was been there with the help of you. The professor mentor who was been there, like Patricia Woliska, she was the one who was the mentor. And naturally, we had the student, master students and PhD students were there involved with respect to this particular project. But what I would say, this was the work which has been done. We have already written the second part is to apply with the field. For that, there's a second level of a project which is already been submitted to European Union. We'll see how far it's going to work, whether we are going to get the grant or not. We have already applied for that. So to do any kind of a research, it simply says that you need a fund. So therefore, I'd like to acknowledge all the funding agencies, even like Nicholas Copernicus University, where I could carry out this work with under Polonus Base Fellowship, where the fund was provided by the NCN and European Union were concerned with respect to providing the fund. The work with respect to nanoparticle synthesis and their optimization was initiated in Amravati University, Santa Gadge Baba Amravati University Biotechnology Department. So naturally, I would like to thank them, even all the funding agencies. And right now, I'm working in Institute of Chemical Technology. So even the things have been acknowledged and with respect to these funding agencies. Mainly, I would like to acknowledge the course coordinator, Professor Manisha Kodape, who's a good researcher who keeps on working. And naturally, we keep on always interacting and discussing with respect to the kind of research which we do. So it's naturally, I still remember when we were in the masters, there used to be a watertight compartment. Like a biology people should not work with chemists and chemists should not work with physicists. But I would say with course of a time, there's going to be a dissolution of these walls which have been there. And nowadays, it's the time of interdisciplinary research. So naturally, I need to always interact with the people from chemistry background, which has been required. So even if I want to prepare any kind of a complex, I have my one particular approach from biological point of view, but there is always a need to have another approach, and that another approach is from the chemist point of view. So I would like to thank the course coordinator, Professor Manisha Kodabek, who gave me this opportunity at least to share my research work and some of the concept related with bio-nanocomposite formation and its application. There are many applications as such which have been there, but this is the one application which mainly we could execute. So just to conclude with the kind of things which have been there. So the way we started with what was our title of a talk, bio-nanocomposites for sustainable development. So we can say that bio-nanocomposite preparation is a green and sustainable approach. Bio-nanocomposite show an increase in total chlorophyll content when given with the maize plant treatment. The treatment was given for 4 weeks of a time. It also, bio-nanocomposite shows plant growth promotion in maize plant in terms of root length, shoot length, and fresh biomass weight, and even the dried biomass weight. So in the end, we are just trying to correlate with respect to the title which we started with. We started with respect to the title, Bio-nanocomposites for Sustainable Development. And we are concluding in the end that this is a green process. In between, we have already explained how we could prepare the nanoparticles and how we could prepare a bio-nanocomposites which were being prepared, and those were naturally, they needs to be characterized from different techniques to understand actually the same composite is being formed or not. So naturally, I thought there will be a question in between, but probably all are mostly the chemical background and they might be knowing all those things. So they didn't have any query. That's good enough. So, but still, if you have any question, I'll be happy to take any particular question. We are well within time. We still have 5 to 6 minutes, which have been deliberately been left for the final discussion, which is being. So any
Sir,
question?
I have one question, sir.
Yes. Mithilesh, Dr. Mithilesh, please go ahead.
Sir, anyway, your lecture was very good, sir. Very informative. Thank you for this lecture.
Thank you.
Sir, actually, you use the zinc nanoparticles with casein many places for your application purpose. I think zinc it's a toxic. Generally, in many papers, researchers provided. What about it's my suggestion, Professor. What about if we use cellulose nanocrystals with casein? How will be this? Have you think about this type project in future or?
First, I would appreciate your question because it's concern. It's important aspect to look at the toxicity aspect of all these nanoparticles. There are reports with the toxicity, but another constraint which comes with the toxicity is the concentration at which you will be using. In case of the complex which we have formed here, the zinc nanoparticles have been used as a source of a zinc ions, which
not audible, sir.
We are not audible, sir.
of micronutrients to a plant, so the concentration is very less, and due to less concentration, because I would say one of the important aspect we miss out with the toxicity study is the concentration. Anything in excess is toxic, so it doesn't mean that we cannot use anything. We are going to use it everything, but within the allowed limit, if you are going to use it, it's always beneficial. So
Okay, Professor. Okay, thank you.
Hello, sir. I have a question.
Yes, sir.
Ganesh Hegde from Karnataka.
Yeah.
Nice work and also a very neat presentation from your side, sir. Sir, one thing I want to know is what do you mean by a controlled release? One thing in your nanocomposites, and also there is another kind of fertilizer like EDTA complexes, like zinc EDTA complex fertilizers. So they also claim there is a controlled release or a slow release of these things. So did you ever check with your nanocomposites with those complex fertilizers?
Yeah, I say with the control release is because I told you I have formed a nutrient which was encapsulated with respect to a protein, where protein is going to serve as a source of a nitrogen. Now, when the plant grows, the root secretes the proteases. The secretion of a proteases is directly correlated with respect to growth of a roots. So more proteases, if they have been released by the roots, they will break down this complex and will release the iron. So more release will be there. If less proteases are secreted, less complex will be broken down and the ions will be made available. So I told you that for control release, the important is the stimulatory response for the release of nutrients. And in our case, with the plant, we have two types of a response. One is with respect to the secretion of enzymes like proteases by the roots when it grows. Naturally, so if it is growing faster, it will have higher requirement. That means it's similar like if you exercise more, naturally your food intake will be more, your body requirement will be more. The same logic which we are trying to apply. So the higher secretion of a protease will make higher availability of a nutrients to a plant due to a degradation of casein and release of nanoparticle, which serves as a source of a nutrient. So we claim this way. Now, with respect to you mention about ADTA, there has to be a strategy. If you say that it is chelated with the ADTA, what is the role they are going to play? Whether ADTA is going to go and get attached with the root surface and will help the accumulation of ions or not, or that particular nutrients which has been there, that needs to be clearly been seen. They will have some kind of a logic to explain how does it works. And many a times we have the advertisement. That advertisement, how far we believe, because they say that tomato is been made from fresh, pure tomatoes, but we know that all those products are basically chemically prepared or chemically flavored. There are no fresh tomatoes anyways in those sauce or other thing. So these two things are different. One, what they claim in advertisement is exactly exists or not. That one is to be verified, and probably you could be the right person to verify and judge the things, whether is it on the basis of advertisement or is it actually has got those kind of ingredients.
Hello.
Yes.
Very nice presentation given by you, sir. It gives, or it will be very fruitful for us.
Sir, what characters distinguish nanoparticles from colloidal particles? This is my first question. Sir, second is we are using transient metal nanoparticles like iron, silver. Can't we use nanoparticles made from P block elements?
You can use it. You can use it. So I think this is Dr. Mohammad Arif. With respect to your question, we can use any other nanoparticle, but you should have some kind of a logic what for you want to use those nanoparticles. Like I have already mentioned, why I use zinc, copper, magnesium, and iron nanoparticle, because all these four nanoparticles serve as a source of a nutrients to a plant. So if you have that kind of a logic with the application, probably you can use it.
Sir, you discuss about the technology transfer
Yes,
and
yes.
biogenic silver nanoparticle.
Could you please, Dr. Bhaskar, could you repeat the question? I could not get the question.
You discuss in your presentation regarding technology transfer and biogenic silver nanoparticle use. So my question is that if this technology is successfully commercialized, what would be the long-term impact on healthcare and that antibacterial resistance?
I'll like to mention here with respect to silver nanoparticle, we say that there is less possibility of developing a resistance. I would like to give a simple logic. There is a possibility to develop, and which is already been seen, to develop a resistance against the antibiotic. The simple logic is that antibiotics has got the specific mode of action. So actually each organism would try to make it survival. Now bacteria wants to survive. So there's going to be evolution come into a play with respect to their survival. So by using antibiotic, all the bacteria are not going to get killed. Some of the bacteria tend to develop a resistance. And it's quite easy to develop a resistance because they know how the mode of action of antibiotic will be there. In case of a silver nanoparticles, the mode of action is not by one means. It kills the bacteria by multiple mode of action. So there is less probability of developing a resistance against silver nanoparticle as compared to antibiotics.
Yes.
So that's the advantage which is there. But this is with respect to the present study and the whatever study which we have done. Still there is work carried out to understand. But right now the advantage is since it kills by multiple ways, even bacteria blocks one of the way, bacteria will get killed by another way. So that way, it has become difficult for a bacteria to develop a resistance against silver nanoparticle, whereas it's quite easy to develop a resistance against antibiotic.
Yes, sir. Thank you.
Yes, anyone else having any questions or the queries? Sir, I think there are no more questions. So if you could permit, shall we conclude with the session, sir?
Okay, as you wish it, there are no questions, no issues. You can share even the email address with that, or probably my email address was been given on the slide. So it's possible anytime if you have any particular question, you can contact and naturally you can communicate. So naturally, not necessary you'll have questions right now.
Thank you, sir.
Ankit,
Thank you.
please,
Thank you,
please
sir.
go ahead.
Now may I request
Thank you so much.
Dr. Sahu sir to please propose vote of thanks for current session. Dr. Sahu sir, please.
Okay, sir. Thank you. On behalf of UGC, MMTC, SGBAU, and all the participants, I, Akash Kumar Sahu, Assistant Professor from Chhattisgarh, would like to extend our heartfelt thanks to our resource person, Dr. Aniket Gade sir, for today's first session on the topic Bio-nanocomposites for Sustainable Development. Sir, your engaging presentation and interactive approach made the session very accessible to all the participants. We truly appreciate your efforts in sharing your valuable insights. Thank you for explaining all the concepts so clearly in your presentation. It was really helpful for us. Thank you so much, sir, once again for this wonderful session. Thank you, sir. Over to you, Ankit, sir.
Thank you, sir. Thank you so much. And with the kind consent of Dr. Gade sir, I would like to declare that this particular session will be over here and will continue for the next session. Thank you so much, sir.
Thank you all for your patience hearing and tolerating with me for 1.5 hours. Thank you all.
Thank you, sir.
I'll leave the, if ma'am permits, I can leave.
Yes, sir.
Okay, thank you.
Okay, so participants, we will move for our next session. I think Dr. Rohan Meshram sir is there. Hello, Dr. Meshram sir.
Yes, sir. I am here.
Yeah,
Hello,
sir.
sir.
Very good afternoon. Very good afternoon, sir.
Good afternoon, sir.
Good afternoon.
We have taken, sir, 5 minutes more from your allotted time. So extremely sorry for that, first of all.
No issues. No issues.
So now may I request Dr. Pogane sir to please introduce our guest first, and then we will start with the session. Yes, doctor, please. Dr. Akulom.
Yes, sir. Sir, am I audible?
Yeah, sir. You are clearly audible.
Okay, thank you so much. Respected our resource person, Dr. Ruhan Janardhan Mishram, and our program organizer, Sir Angit, and our course coordinator, Professor Dr. Manisha Gutabe, and my fellow STEAM participants. A very blessed good afternoon to you all. This live season topic will be on molecular dynamics simulations for chemistry teachers, concepts, applications, and demonstration using GroWin, which will be delivered by our resource person. So before he deliver his topic, let me give a short introduction of our resource person. His name is Dr. Ruhan Janardhan Mishram, and he is an accomplished and a distinguished researcher in bioinformatics, computational biology, molecular modeling, and artificial intelligence-driven drug discovery, presently serving as an assistant professor at the Bioinformatics Center, Savitribai Phule Boni University, Boni. With more than 19 years of teaching, research, and academic administration experience, he has made significant contribution to the advancement of computational approaches in life sciences through interdisciplinary research, software development, and capacity building. He holds an MSc and PhD in bioinformatics, has qualified to SET in life sciences, and is a recipient of the prestigious Indian Council of Medical Research postdoctoral fellowship, reflecting his excellence in biomedical research. Dr. Mishram's research encompasses structure-based drug design, molecular talking, molecular dynamic simulations, computational medicinal chemistry, viral bioinformatics, reverse vaccinology, artificial intelligence applications in bioinformatics, and the development of bioinformatics software, databases, and well-based computational resources. His work bridges computational and experimental biology, enabling the identification of novel therapeutic targets and accelerating drug discovery through advanced computational methodologies. He has successfully secured and led high-value externally funded research projects from premier national funding agencies, including Department of Biotechnology, Department of Science and Technology, and Anusandan National Research Foundation, having collectively valued at over rupees 700 lakhs. With this milestone, he has significantly strengthened research infrastructure and computational biology capabilities at Savitribai Phule Pune University. Also, as an internationally recognized scholar, Dr. Mishram has authored 65 research publications in reputed international journals published by Elsevier, Springer Nature, Wiley, Deller, and Francis, and Nature Publishing Group, with over 1,430 citations and an H-index of TIND. His research has contributed substantially to computational drug delivery, infectious diseases, cancer biology, medicinal chemistry, and molecular simulations. He is also an innovator with multiple international patents, published Indian patent applications, software copyrights, scientific databases, and standalone bioinformatics software, those that are widely accessed by researchers across the globe. As an academic mentor, Dr. Mishram has successfully guided 47 postgraduate dissertations, and he is currently supervising doctoral scholars and has actively contributed to curriculum development, syllabus revision, and postgraduate program design in bioinformatics and biotechnology. He has also delivered numerous invited lectures, keynote addresses, faculty development programs, refresher courses, and national workshops on emerging areas in life sciences and chemical sciences. His commitment to academic excellence is further reflected through his active involvement in university governance, research advisory committees, examination reforms, student mentoring, and various institutional responsibilities. And with a strong blend of research excellence, technology innovations, academic leadership, and interdisciplinary collaboration, Dr. Rohan Janardhan Mishram continues to contribute significantly to advancing bioinformatics research, promoting computational approaches in biomedical sciences, and developing next-generation researchers capable of addressing contemporary challenges in healthcare and life sciences. Sir, with all your years of experience in research and teachings, we are honored and intrigued to have you here and to be able to learn from you through this program. With this, I conclude my introductory part, and I give the remaining then to our resource person, Dr. Rohan Mishram, to deliver his talk. Thank you, sir. Sir, you may kindly show your slide.
Okay, Dr. Punjan, thank you very much. I really don't know if I really deserve half of the things that you have said. I really appreciate your kind words. I hope I am audible as well as visible. Before, without any ado, let us begin, because we have already exceeded our time. Okay, I hope I am allowed to share my screen. Okay, now is my screen visible to you guys?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
Yes.
It is always kind of suspense if things work or not every time, so please don't mind this thing, right? Okay, so my friends, this talk is going to be really, I don't want to be any formal practice in this thing. I want it to be really very informal. I want it to be really very interactive. Okay, so I would need a lot of you can say input from you people, because the talk is really not about when it is said ki I am going to talk about molecular dynamics and simulation. So be assured that I am not going to bore you to death by going into mathematical details of all those expressions and things. Okay, so we are going to talk about molecular dynamics in terms of teaching and in terms of training, in terms of getting it up and started. Right. So first and foremost, I would like to have just a brief outlook uh, as to how many of you have used computational techniques in your teaching or in your research as such. You can write it in the meeting chat, or you can just share your experience कि आप लोग क्या क्या use करते हैं when it comes to computational things in your own research and teaching. Okay, so what maybe what software do you use? Feel free to talk. Okay, so Alok Verma is saying that he has used Autodoc, and Sudhanshu Pratap Singh is saying that he has not used anything so far. Okay, while someone has used DFT, someone has used CameDraw. Okay, so you are not sure. So I see that some people are using it for DFT, right? So this is good. Okay, so I see yes, image J, Avogadro, CameDraw. Okay, fine. So okay, so I get it. Okay. So now let us talk some another question that I want to. Okay, so how many of you have used MD simulation in your research or training or teaching? MD simulation किसी ने use किया है क्या? Okay, and if you have used, what software have you tried? Okay, yes, there are people like GroMax. Yes, this is good. Ankita, you will enjoy today's talk then. Then Sudhanshu says he has not used. Okay, and anyone else? Okay, so if that is the case, if you are okay, Abhishek says that MD simulation used in research by my collaborator. So that is usually things happen. People from chemistry background, they tend to collaborate with some computational biology person and let these guys do the work. Okay, that is really very fine. And okay, so can you tell? So whatever people or whoever people have not used MD simulations in their teaching or research, what is the reason that holds you back? Yes, like Dr. Salim Marin says, MD tools need high-end workstation. So that might be one of the limiting factors कि लोग ज़्यादातर MD simulation के पीछे नहीं लगते. Well, we are going to address this issue, my dear. Okay. So are there any other reason that has limited you or your students to use MD simulation in your training or research as such? Right. Need of high-end workstation is one of the major factor. Yes, of course, it requires cost. If you are Alok sir, yes, you are on the very right answer. So cost is another thing. Okay, it is time consuming, of course. Mitali Sen, yes, you are right. It requires a lot of time. The calculations are quite really very long. Okay. So you also need some theoretical knowledge and interest to do that. Yes, you might have to grind yourself a little bit in order to get it up and started. Okay, so these are some of the challenges that the scientific community, and by scientific community I mean chemistry scientific community, are dealing with when it comes to implementation of MD simulation in their research and teaching. Okay, so being said that, well, now today's talk we are going to deal with like how we can overcome these challenges. Like what are the solutions that are available? Of course, we would start out with what exactly MD simulation is, and why is that chemistry people might want to use that thing in your research. Okay, so in terms of chemistry point of view, in terms of chemistry research or in terms of chemistry training. It may be your PhD student training, or it can be your MSc student training. Okay, so this MD simulation basically chips in where you deal with synthesis of small molecules. If you are dealing with phytochemicals, novel chemical compounds that you people usually synthesize in your lab. And after you complete your testing against, say, some cell-based assay, MTT assay, or it can be enzyme inhibition assay, then you might come to a point where you might start thinking as to how exactly that my compound that I have created might have resulted in the anti-cancer activity, or what might be the structural reason that would satisfy my observation that my compound is inhibiting certain enzyme, and so on and so forth. And in that context, my friends, this molecular docking and MD simulation comes into play. So usually what people do is that they end up docking the compounds that they have generated with, say, some therapeutically important protein target. Right. So usually these targets are protein molecules, and what they end up is like I have a protein molecule, and this is how my ligand would orient in my protein. Right. So this is where the docking system ends. Okay, so now, my friends, let us talk about why is that people want to proceed one step ahead when it comes to analyzing the protein ligand interactions as such, right? So, what are the motivation of doing these MD simulation? Of course, publication is one of the motivations. Nowadays, if you simply have your docking data, you might be able to publish it in, say, some decent journal if you write it properly. But if you have MD simulation data in addition to molecular docking data, then maybe you can try higher impact factor journal that would, of course, get you ahead in your career, right? So besides that, getting MD simulations into your research, well, it will give a detailed idea of including the concept of time dimension into your study, or time dimension in your interaction analysis as such. So by that, I mean that whatever proteins that you might have downloaded and docked with your ligand molecule, or with your newly synthesized compounds. So once you get docking, it seems that you have a static condition. Okay, this is my protein and this is my ligand, and it seems to be static. But proteins or enzymes or your drug target, my friend, at physiological condition, and even when you test them in your laboratory under in vitro conditions as well. So these are not static as they appear in the docking result. But these protein ligand complex are in constant state of dynamics. So these entails motion in these systems. So there is always motion in your protein ligand complexes, and these motions can be local. For example, there can be a flexible loop in your protein that can move a little bit, right? Or there can be side chain rotation in your binding site or active site, or these motions can be in form of global rearrangement, like entire domains can be rearranged. That can be crucial for proteins functions and physiology. So eventually a lot of motions decide how your ligand is going to interact with your protein, or how your newly synthesized drug is going to interact with your protein. So the time scale of the motions are also important. So there are some fast motion that can happen in your system. Now, by system, I would refer it to your protein ligand complex, right? So fast motion happens in your system that can be in range of femtosecond to nanosecond. Okay, so this involves bond stretching, like angle bending. It can involve side chain flipping. So these happens in the range of say femtosecond to nanosecond. While the motions can also be slow. So the slow motion occur over microseconds or even milliseconds that involve collective motions of many atoms, such as rearrangement of secondary structural elements or tertiary structural elements at the larger scale as such, right? So now let us talk about what are the experimental techniques that are used for monitoring motions in biomarkers. Okay, so it is not like we directly jump on to computational thing before we have any experimental evidences, right? So there are options like you can monitor motions in biomolecules using this ultra resolution X-ray crystallography. You can use ultra fast NMR. You can use AFM for your motion monitoring. Sometimes fluorescent spectroscopy can also be implemented. But there are certain limitation of this experimental technique. Most of them are really very expensive. They of course need cutting edge infrastructure to implement such experimentation, and it requires highly trained human resources. So the shortcut to all these things is that you can use computational approach to monitor the motions that are happening in your drug and protein molecule that you have generated. Okay, so computational includes approaches for monitoring the motion may include MD simulations. That is going to be the major major focus of our talk today. Or there are also hybrid QMMM calculation which you can implement if you are trying to deal with breaking and making of bonds and so on and so forth. But most of the time in this lecture, we are going to talk about MD simulations, right? So to begin with, let me tell about what exactly is the basic prerequisite for conducting the MD simulations, right? So for beginning any MD simulation, you might need the particle-based description of your system. So this seems to be a very big word, but just relax, my friends. It is a very easy concept. Okay. So this particle-based description means, well, it is the very first step, you can say, to construct a particle-based description. It would tell as to how many atoms are there in your protein, and most of these atoms are represented by atomic coordinates, and these atoms are connected to each other by bonds. Okay, so let me basically show you how is that you define the particle-based description. So as you can see, my friends, you can consider this as the particle-based description of a protein system that the protein that you are visualizing right away. It is aldose reductase that is in a complex with say some inhibitor, flavonoid inhibitor. I will just show you. This is the inhibitor, and the ribbon representation that you are watching along is the protein molecule, and this small lines that you see in, well, this is the small molecule inhibitor that we have maybe designed. Okay, so as you can see, now I will turn off the ribbon representation and show you in lines. So what you can see here, my friend, is the particle-based description of your system, wherein each and every atom that you see, this red one that you see is an oxygen, the white one that you see is carbon, blue ones that you see is nitrogen. So each and every atom, well, we represent them in form of three-dimensional coordinate with respect to say some frame of reference, right? So this is how you should begin. इसके बगैर आप MD सिमुलेशन शुरू नहीं कर सकते. आपको सबसे पहले पता रहना चाहिए कि what exactly our system is, how many atoms are there, okay? And usually it is an easier thing to do. You can download the initial system right from PDB, or you can generate homology model, right? So if you download it from PDB, or if you this particular system that you are watching right away, it is we have obtained it from docking results as such. This is an artificial compound that is in complex with this protein molecule. Okay, so this is how you kind of create a particle based system. Okay, so next thing, once you have this input with you, this basic input, the main idea of MD simulation is to propagate this system in time, right? So now what you are supposed to do is to propagate this system in time, well, say for say some finite time. The idea is that how my system would behave if I propagate in time naturally for say 100 nanosecond, for say 500 nanosecond, or even if you now with the availability of high throughput, or you can say HPC clusters available, people have stretched these timelines to even microseconds and milliseconds. Okay, so this is what we do. So what you have in docking result is this, a static picture. Now by using MD simulation, you would be able to propagate your system in time and see how my system behaves, how my ligand would orient itself in the binding site of my protein. Can it form new hydrogen bonds, or can it form new interactions that were missed during docking? So that is where this MD simulation comes into play. And when you kind of propagate this system in time, we say that you have simulated your protein ligand system. That is what people usually refer to when we say that we are simulating the system, or when we are simulating the protein ligand complex. They actually refer to say, okay, we are trying to see how it behaves with respect to time. Okay, now how is that you decide the time span of your simulation? So duration of the simulation, that is, if I want to propagate my system for nanoseconds, or if I want to propagate my system for even microsecond, मैं कैसे क्या decide करूँ इसको? So my friends, इसका जवाब it depends on what is the research question that you are trying to address. For example, if you have newly synthesized a small molecule in your laboratory and your objective is to just figure out if my ligand, or if the new molecule forms a stable complex with my protein or not, and you usually do that by calculating the binding free energies. And for calculating binding free energy, a small for a small time scale of nanosecond are small time scale of simulations are sufficient. Let's say, for example, typically 50 to 100 nanosecond simulations are sufficient to answer this question. So, but if your objective is to monitor how conformational changes are affecting my small molecule, then you might want to extend your simulation till well, say for example, microseconds or even milliseconds. Okay, so most of the time that I have dealt with the medicinal chemist or drug chemist, 50 nanosecond to 100 nanosecond simulation would be more than enough. That is what they come up with, right? Okay, so now you know कि अपने को कितनी देर तक simulate करना पड़ेगा अपने system. So every molecular dynamics simulation, or you can say every simulation, that usually results into a trajectory. तो हर एक simulation का end point होता है trajectory, और ये trajectory is, you can say, nothing but trajectory is nothing but it kind of records like what things are happening in your system over time. Okay, so maybe I will show you a trajectories so that things might become more clear. So this you can see, my friends, is a system wherein जो बीच का portion है, the blue color that you see, that is a protein molecule, and we have kind of placed that protein molecule in a water box, and we have simulated it say for 100 nanosecond, and this is the trajectory that I am going to show you. I have loaded the system in VMD program, and if you run this trajectory, now you can see with the passage of time, there are motion in your system, right? And this is something called as the trajectory, right? So over time period, this is how my system has evolved. This is how the changes might have happened in my system. So in this trajectory, now you can see, जो यहाँ पर change हो रहा है, it is the frame number. So trajectory is composed of various frames, right? So what I will do, I will slow down the speed of the frames in my trajectory and again play it. Now you can see the frames in my trajectories are moving slow. Okay, so well, say for example, this is a 100 nanosecond simulation and I have 1000 frame. That means there is the time lapse of 100 picosecond between every 2 frames. Okay, so these frames correspond to, okay, after 100 picosecond, this is how my system will look like. After next 100 picosecond, this is how my system would look like. After next 100 picosecond, this is how my system look like. And we would continue this for say 100 nanosecond. Okay, so to give you an idea, maybe I will change the representation so that you can have a better look at this trajectory. I would create a representation for only protein molecule. Okay, and maybe I will change the drawing so that you can, this is how a protein usually familiar with, you are familiar with, right? And maybe then I will show you how is that the ligand looks like in your protein. Okay. So you can use the residue name over here. I know the name of my residue in my system is ligand, and I would create a representation and show this representation in beautiful CPK. Now you can see that this is your ligand molecule. You can see that it is moving along, right? Maybe I will increase the speed. Okay, so in this way, you know, this is a typical trajectory that gives you an idea as to how my ligand would behave in the binding site of my protein. You can see that when you bring time dimension into picture, you can now see that this functional group might reorient and can form new hydrogen bond that might have been missed in molecular docking, so on and so forth. So this is the ultimate objective of, or you can say this is the ultimate end point of any MD simulation. Okay, so now how do we do this thing? Right? ये आज अपन सीखने वाले हैं. So let me quit this thing. Okay. So let us not go into more details of this thing. Let us now talk about what are the software options that are. Of course, there are very standard open source softwares that are available, which include GROMACS and NAMD. And if you tend to use commercial software, then of course there are options available like Schrodinger. It uses Desmond in its background. You can buy BioVia Discovery Studio, which eventually implement CharMem and NAMD in its background. So these are options that are available. So now let us talk about what are the general steps that are involved in MD simulation. So the very first step is that you need to prepare your system, right? So you need to obtain the model, which you can obtain from PDB, or the models that you can begin with can be from molecular docking results, right? Next thing that you need to do is you have to obtain the topologies and parameters of ligand. So you can use softwares like SwissParam and CharmM GUI to obtain the parameters. We will come to what exactly are parameters and topologies in a while, right? So these are the preparatory steps. So once you have this thing in your hand, then you can begin with system preparation. You usually end up with your system in PDB format. But when you use MD simulation software, you would need input that are recognized by those software. So, well, say for example, if you are using NAMD, then you might need the input in PDB PSF pair, or if you are using GroMax, you might need the inputs in Gro file format as such. Okay, so once you have initial structures in software readable format, then you can proceed for solvation. The solvated protein that I have shown you, that you can place your protein in a water box, it would look really very beautiful, right? And then it is possible for you to neutralize the charges, or you can say by adding certain ions, if there is if the charges are disturbed, then you can add specific amount of chloride ions or specific amount of sodium ions in order to neutralize your system. So you can perform charge neutralization. That is what you usually do. And usual CMD implementation, after doing this thing, when you have your system ready, you first perform minimization, which relieves the bad contact and it lowers the system potential energy. And of course, you would have to adjust your system to force field. अभी force field क्या है, इसके बारे में भी अपन थोड़ी देर बाद चर्चा करेंगे. So once you have made sure that there are no bad contacts between my protein and water molecule, next thing that you do is that you slowly increase the temperature of your protein ligand system in that water box to a physiological temperature, we'll say for example 300 Kelvin. Okay, so once you have brought the system to physiological temperature, room temperature, while doing so, some kinetic energy is pumped into your system, and now you need to stabilize the system under normal temperature and pressure conditions as such. Okay, and for that we usually perform equilibration, and after equilibration, you usually collect the data. Now once you have performed equilibration, that means you have stabilized the system under normal pressure and temperatures. Now begins the final and most important thing, to propagate your system in time. That is what happens in your production run. So in this production run, you usually collect the data, right? So the trajectory that I showed you, the movie that I showed you, that was from the production run for topoisomerase 1 from Leishmanian in complex with certain small molecule that we are working with. Okay, so now let us talk about कि मैं आपको अभी क्या demonstrate करने वाला. Okay, so we are going to deal with the software GroMax, and most importantly, we are going to deal with the Windows version of GroMax that we have kind of tried to develop at my lab at Bioinformatics Center. We call it as GroWin. तो ये जो GroMax सॉफ्टवेयर है, ये कहाँ से डाउनलोड कर सकते हैं, ये GroWin सॉफ्टवेयर कहाँ से डाउनलोड कर सकते हैं? Okay, so of course you can download it. It is of course free. You can download it from SourceForge website. SourceForge website is a public repository of open source softwares. वहाँ पर आप अगर GroWin करके सर्च करेंगे, you would end up with this first page, right? And in this file section, you can have sorry, it is taking some time. Okay, so in this file section, you can download either the GPU version or the CPU version. We have kind of created these two separate versions. So most of the time, for anyone who is working out of bioinformatics or computational biology domain, the CPU version is what you might want to start out with, right? So which version is suitable for me, GPU or CPU? Of course, if you are confident that I have GPU in my machine, तो GPU यानी एग्जैक्टली क्या, you can say कि if you have specifically purchased any graphics card, right, NVIDIA का ग्राफिक्स कार्ड अगर आपने खरीदी करके उसको इंस्टॉल किया है आपके सिस्टम में, then you can say that your system has GPU. तो यहाँ पर थोड़ा कंफ्यूजन हो जाता है इनिशियली लोगों में कि भाई there are inbuilt GPU cards. Okay, so they are not fine tuned to work with molecular dynamics softwares like GroMax and NAMD. आपको अलग से GPU कार्ड खरीदना पड़ेगा, ठीक है। NVIDIA का खरीदोगे तो that are most compatible with all open source MD simulation softwares like NAMD and GroMax, right? तो अगर आपने GroMax अगर आपने GPU कार्ड खरीद कर रखा है, then go for this GPU version, or CPU version will work if you are not sure about it, right? Okay, so now what are the prerequisite to use this software? ये software use करने का prerequisite क्या है? सबसे पहली बात तो you might want to first install this Microsoft Visual C++ redistributable, which is of course freely distributed by Microsoft. ये आपको download करना पड़ेगा from the Microsoft website, right? So once you download it, this is how you might look like. यहाँ से आपको you click over here. These are the two executives that you need to download. तो अगर आप इसको download करते हैं, तो this is how it might look like. Just for okay. ये आपको पहले डाउनलोड करके इंस्टॉल करना पड़ेगा, इसके बगैर आपका काम आगे नहीं जाएगा, राइट। नेक्स्ट थिंग दैट यू आर सपोज टू डू इज दैट अगर आपके पास में GPU है तो आपको पूरा वहां पर इंस्टॉल करना पड़ेगा, ओनली इफ यू हैव GPU, राइट। तो GPU अगर आपके पास में नहीं है तो डाउनलोडिंग दिस माइट वर्क. Operating system, we have tested this software on Windows 8 and I would recommend कि आप Windows 11 या फिर उसके ऊपर use कर सकते हैं. The system that I am using now is Windows 8 system. Okay, so now how do I install it? Installation भी काफी आसान है. We have created a small इंस्टॉलर दैट यू कैन डाउनलोड. यहाँ से अगर आप इंस्टॉल करते डाउनलोड करते हैं इसको CPU वर्जन, दिस इज हाउ द फाइल विल लुक लाइक. ऑल यू हैव टू डू इज टू राइट क्लिक इट and make sure that you run it as administrator. Okay, and once you run it as administrator, it can maybe ask you if you want to run it. Okay, so we will go with the English as a default. Okay, here itself it will tell you what are the system requirement that are there. Okay, make sure that you have done everything. Hit next. This is the rate limiting step. आपको ये change करना पड़ेगा. By default it will install your program into this program files x86. आपको इसको change करके you have to install it into program files and hit OK and then proceed for your installation. After the successful installation, I have already done this thing, so I will exit the setup. You might end up with something like this. In your C drive, you might end up in program files. There would be a folder called as GroMax. ऐसा होना चाहिए. Otherwise, your software will not work. And after successful, installation, you will get this shortcut for GroMax, and there you go. Double clicking it will launch your GroMax program. So in this way, my friend, we have kind of crossed the platform barrier. तो लोगों को ज्यादातर इनिशियली ये तकलीफ होती है कि सर आई डोंट have that Linux system installed on my system, so I'm finding it difficult to do that. So by using the Windows version of GroMax, of course, that particular barrier we have already gone through, right? So now let us talk about how do we use this thing. Okay, so for training your students, okay, we have kind of created a simple protein ligand tutorial. Okay, जो यहाँ पर जब आप जाएंगे, if you click on this particular link, you will get all the pre-required data that is required to run a simple simulation, right? तो आपको ये सब चीजें डाउनलोड करनी है, and once you download, this is how things might look like. तो अभी मैं बताता हूँ आपको कि ये ट्रेनिंग किट में exactly क्या है. Okay, so in this training kit you have ligand parameterization data, जिसमें आपके ligand के coordinates है और उसको लगने वाली ITP फाइल्स है. All the MDP files that are required to run the simulations that are there in your toolkit, like em.mdp, md.mdp. What are these MDP files now? These MDP files are nothing but these molecular dynamics softwares like GroMax. It kind of needs a script that will guide it as to how do you run the simulation. Right. So these MDP files are, you can say, the blueprint that will tell GroMax how to run the simulation. Okay. So I have also created some pipelines that can be used to automate the workflow. Just a minute. Hello. Oh. Okay. Huh. And generally, kids, when you try to train them, they end up with like, sir, I'm having difficulty memorizing the GroMax command. So to do that as well, to deal with that as well, we have created this small file wherein you can simply copy paste the commands that are required. राइट, तो आपको टाइप करने की भी जरूरत नहीं पड़ेगी, आपके बच्चों को टाइप करने की जरूरत नहीं पड़ेगी, सिंपली कॉपी पेस्ट इट एंड सी हाउ थिंग्स हैपन. ओके, सो देयर इज आल्सो अ ट्यूटोरियल which you can see for help. Okay, wherein we have provided how is that you generate everything with screenshot of how things would look like on your screen so that you might not get lost when you run those commands. Okay, तो ये आपकी मदद के लिए है, ये आपके बच्चों की जिन्हें भी आप ट्रेन कर रहे हैं. We have tried to make it as simple as possible, right? The main idea is to prevent or to get around that entry barrier that faculty and student usually have when it comes to MD simulation. Okay, तो इससे भी आगे we have our own YouTube channel जहां पर whatever I am going to demonstrate you right away, उसका ऑलरेडी हम लोगों ने YouTube पर वीडियोस बना कर रखे हैं. You can see them if you get lost during this particular workflow. Okay, so this is how things are, my friend. Okay, so now let us begin. तो पहली स्टेप होती है, how much time do we have now? अपने पास में और कितना टाइम अवेलेबल है?
Hello, sir. I think 30 minutes.
We still have 30 minutes, right? Okay, so is there any question? So far so good, because I have been going on talking continuously. अगर नहीं होगा तो maybe I will demonstrate you and then you can ask.
Sir, please continue.
Okay, fine.
Yes, sir. You can demonstrate.
Okay, so things are quite really very simple. We have come most of the things halfway through. Okay, तो अभी करना क्या है आप? Once you have this GroMax installed and ready to run, what you are supposed to do is to change the working directory first. Okay, so by default the working directory is somewhere in C program files, and all my input data is somewhere in D drive in say some folder GroWin. तो आपको क्या करना पड़ेगा पहले? You need to change the working directory and reach where all your input files are. Okay, so it is quite really very simple. All you have to do is to go here You need to copy the location and give the simple see the directory command, and maybe paste it over here, or it will not work this way. You need to change the directory first. When we begin, we are in C directory, all the data is in D drive. So we need to change the drive, then give this cd command and provide the path where our input files are. You can use this dir command just to make sure that all the files that are there in your folders are listed over here. So this would give you a kind of confirmation कि आप सही folder में, right? Okay, so now you are in the right folder. The very first thing that you need to do is to obtain the GROMACS readable initial structure. So इसके लिए GROMACS uses commands. Like there is PDB to GMX command that generally convert your receptor molecule that is there in this receptor file into GRO file. That is, read by GROMACS as such. तो उसके लिए आपको कौन सी कमांड रन करनी पड़ेगी? वी आर गोइंग टू यूज दिस PDB to GMX कमांड. तो ये PDB to GMX कमांड में आप फोर्स फील्ड सिलेक्ट करते हैं. In our case, we are going to use CharMM27 force field. तो GROMACS also offer other force field जिसके बारे में अपन यहाँ पर अभी चर्चा नहीं करेंगे. तो for our case, we are going to use this CharMM27 force field, and the water model that we are supposed to use is TIP3P, right? So these things are to be accomplished through this PDB to GMX command. So how is that you proceed with ये जो GROMACS command का जो folder है, ये जो GROMACS command का जो file है, इसको अपन open कर लेते हैं. All the commands are already here. All you have to do while training your student is to open this thing, copy this command, okay, go to your console and paste it. So this command says GMX is something that will invoke GROMACS in the background. PDB to GMX is the command that will tell that it needs to convert the receptor file that is here in PDB format into GRO format. Okay, -F is a flag that tells that the input file is in the file is with the name rec.pdb जो यहाँ पर आपके इसमें रहना चाहिए. Okay. And -IGNS says that ignore hydrogen. So this we usually do it, right? So once you do it, once you copy and paste the command, just hit enter. Okay. Now your GROMACS is asking you what force field that you need to use. Okay. So as I have already told you, we would be using CharMM27 force field, which is present on 8th option. So we would provide here option number 8 and hit enter. Now it is asking what water model that you are interested to use in the simulation. So we would be using the first option, TIP3P. It is recommended by GROMACS, therefore we are using it, right? So just hit 1. Okay, there is a menu and you are supposed to select something from this menu. Okay, so we would be using this TIP3P model, therefore use 1 and hit enter. And that is what my friend. Okay, so after successful completion of any command, GROMACS will give you this code, like GROMACS remind you, and there would be certain code. And if you end up with this thing, that is an indication that you have successfully completed the task. Otherwise, it will throw some error. Okay, now you can see if I sort by date modified, it has recently generated these 3 new files. Right, the 3 new files that are generated are ये जो top file दिख रहा है आपको, it is a topology file. ये एक ITP file है, posre.itp, it is for position restraint. और ये जो है आपका conf.grow, इसको अगर आप properties देखेंगे इसकी, तो इसका extension है grow. It is a grow file which is now readable by GROMACS software as such. Right, तो ये 3 फाइल अभी PDB to GMX में generate हुई. Okay. ठीक है. So now next thing that we need to do is that we need to prepare the ligand. So abhi we have dealt with only the receptor, right, the protein molecule. But now we also need to have your ligand. ये आपके लिए ज़्यादा relevant रहने वाला है, because the ligand is the thing that you would synthesize in your lab. ठीक है? तो GroMax को प्रोटीन के बारे में मालूम है. It knows everything about protein. What are the amino acid residues that are there? What are the atoms that are present in your protein molecule? How are they connected to each other? But the ligand that you would be synthesizing, that would be new, right? तो आपको ये भी बताना पड़ेगा ग्रोमैक्स को कि भाई हाउ इज दैट माय लाइगैंड इज, राइट? तो उसके लिए आपको आपका लाइगैंड प्रिपेयर करना पड़ेगा, राइट? सो टू प्रिपेयर द लाइगैंड, फर्स्ट यू नीड टू कन्वर्ट योर लाइगैंड इनटू ग्रो फॉर्मेट, राइट? तो उसके लिए दिस इज द कमांड, editcunf कमांड रहती है ग्रोमैक्स में, दैट वी आर गोइंग टू यूज, राइट? सो मेक श्योर दैट यू सिंपली कॉपी एंड पेस्ट इट ओवर हियर. The input file is leak.pdb, that should be there, यहाँ पर already है. Make sure कि ये सारी चीजें वहाँ पर रहनी चाहिए. And the output would be leak.grow. Okay, and once you have copied and pasted this thing, just hit enter. और यहाँ पर देखो leak.grow नाम का एक नया फाइल तैयार है. Now your protein as well as ligand both are there in GroMax readable format. Okay, so that is what we have done. Okay, so the next step that we need to do now, कि अभी problem ये है कि both of these files, they are separate, but we have the complex of your protein and ligand, right? So we kind of need to merge this information in one place, right? So you can use any text editor, like I'm using this notepad++. अगर आपके पास में notepad होगा वो भी चल जाएगा, कोई दिक्कत नहीं है. Okay, so now what you need to do is that whatever lines that starts with one leg, उसको आप copy करना है. Whatever lines that start with this ligand, okay, all you have to do is go to edit and go to copy. Okay, और ये आपका प्रोटीन का इंफॉर्मेशन है. You can see that the first residue is alanine, so on and so forth. All you need to go is to scroll very back down. Once you reach the end of your protein molecule, that is, this is the last atom of your protein molecule. Okay, just hit and enter and go to edit and paste it. Okay, so what you might want to do is that you might want to remove this excess of 2 spaces that have appeared over here, or maybe we will remove first one. We need to align all these things. That is what we need to do. Okay, things are working. Okay, so make sure that these 3 coordinates of your protein as well as ligands are aligned quite well. Once you do this thing, now save it. Okay, so after doing this, my friends, next thing that you are supposed to do is that we have added the information of our ligand over here. And the ligand information is there in, say, 33 lines, जो आपको यहाँ पर पता चले, right? So initially, the protein information was in this 50 lines. That means there are 50 atoms in your protein, and now you have added 33 more. So you need to add this 50 plus the 33 lines more, and you would have to sum it up. So the sum of the entire atoms would be eventually 83. Right? So this will tell your GroMax that there are 83 atoms in your system now, and it contains protein and ligand. So you need to save this thing. The initial topology file that we have generated, अपने को उसमें भी बताना पड़ेगा कि we have ligand in there. Okay, this topol.top is the topology information. ओके तो आपको क्या करना है कि यू नीड टू लुक इनटू दिस section that include force field parameters like this is for protein right तो अभी आपको इसमें आपके ligand का भी इंफॉर्मेशन डालना है राइट ओके I will copy this. It is already there in your commands file. Okay, and I will paste it over here. So by doing so, you are telling the GroMax software कि along with protein, there is something called as ligand.itp जिसमें सारा topology का information है. Topology यानी क्या? How is that your atoms are connected to each other? What is the bond angle? What is the bond distance? So on and so forth. Okay. So ये एक चीज हो गई. The next thing that you need to do is that you need to upload the list at the very end, right? इसके end में when you go down, अभी यहाँ पर सिर्फ प्रोटीन ही है. Now you need to tell कि there is another component called as ligand in your system, right? और कितने ligand है? There are one ligand. Okay, so this enables GroMax to understand that the system contains one protein and one ligand. Okay, so if there are multiple ligands, then maybe you can add the number of ligands over here. इसको आपको सेव करना है. You need to save this thing. Okay, ठीक है. So now your topology file also has this ligand information. Okay, so once you have done this thing, you need to edit the ligand.itp file. This is the next thing. Hello. Sorry for that. Okay. Now we need to edit some ligand.itp file. It is not necessary, okay, to do so, but still make sure that whenever you deal with the ligand molecule, the ligand.itp file is usually what you obtain from the parameterization step. Okay, so यहाँ पर आपको lig.itp file मिलेंगी. You need to copy this file into your working directory, paste it over here. Okay, and you just have to make sure कि यहाँ पर in this section molecule type name NRXEL there is lig3 written over here. Just make sure. अगर आपने कुछ चेंज कर दिया तो हो सकता है कि यहाँ पर वैल्यूज चेंज हो जाए और आगे चलकर वो गड़बड़ कर सकता है. So it is right where what we need it. Okay, so we don't have to worry about it right now. Okay, so now let us begin our job of placing our protein in the water box. Okay, तो उसके लिए we will be using something called as the creating box. Edit GroMax edit conf कमांड अपन ज्यादातर यूज करते हैं. ओके, तो वो यहीं पर है. ओके, so we would be using this edit GroMax command. Just copy this, right, and paste it in your GroMax console and just hit enter. Okay, and now you can see it has created a new file called as box.gro, right? So box.gro file तैयार होगी. Okay, what we have done now is that whatever protein that we have, this protein इसके आजू बाजू में अपन ने एक बॉक्स तैयार कर लिया है, right? और उस बॉक्स में now we need to populate that box with small water molecules जो आपके प्रोटीन को सराउंड करेंगे. Okay, and for that we would be using GroMax solvent command जो आपको यहाँ पर दिखेंगे. All you have to do is to copy this thing, paste it, and hit enter. Okay, so this will create another file called as boxsol.gro. Now you can open this boxsol.gro in VMD so that you can see what exactly you have done. Okay, तो अभी दिख रहा है आपको? This is the protein molecule that we have started with. ये वही protein molecule है जो मैंने आपको initially बताया था. And now you have placed it in this solvation box, right? अभी एक और चीज करनी अपने को बाकी है before we start the simulation. We need to neutralize the charges or we need to adjust the pH of our system. ठीक है? तो उसके लिए we would be first using the GenIon command. बट जेनायन कमांड यूज करने के पहले अपने को उसका एक टीपीआर फाइल तैयार करना होता है. सो टीपीआर फाइल क्या होते हैं यूजुअली, टीपीआर फाइल आर समथिंग दैट इनेबल्स योर ग्रोमैक्स टू नो कि उसके अंदर क्या आने वाला है. वेल, इसके बारे में अपन थोड़ी देर बाद चर्चा करेंगे, राइट? तो बिफोर यू सॉल्वेट योर सिस्टम you need to create a TPR file जो GroMax में grompp command है. ठीक है? This grompp command is used to create a TPR. Okay. So here we go, my friends. Paste it. It will create a file called as ion.tpr. Okay. Now you can see ion.tpr तैयार होगी यहाँ पर. Okay. Now you can use this ion.tpr file and you can add the ions molecule using this GroMax GenIon command, right? तो यहाँ पर let us use this GroMax GenIon command. यहाँ पर अपन paste करते हैं, right? GMX GenIon, that says कि now we are adding the ion, right? So it will create a file called as box solve ion.grow that would contains the ions that are added. तो ये ion addition का काम कैसे होता है? So now in this previous step that you have generated, over here you can see that there are water molecules. So this GroMax GenIon command क्या करेगा? It would look around in the entire system and see if it needs to neutralize the charges, and then it would eventually calculate कि भाई how many extra sodium ions are needed to be added or how many extra chloride ions are needed to be added in order to make our system neutral, right? So when you hit this enter, it will now calculate the number of ions that are required to neutralize the system, and now it would ask you कि भाई इसमें से कौन से atom आपको replace करने हैं. तो usually we end up we would replace the solvent molecules. जो एक दो जो randomly water molecules अपन select करेंगे and we would replace them with the number of ions that are required. So these solvent are there at option number 15. So we would be using after number 15. So अगर आपने 15 यूज किया तो ये क्या करेगा, कुछ वाटर मॉलिक्यूल रिप्लेस कर देगा आपके सोडियम और क्लोराइड आयन से, एंड योर सिस्टम वुड बी चार्ज न्यूट्रलाइज्ड. Okay, now you can see that it has replaced this much Na ion and it has added this much of chloride ions and so on and so forth. और आखिरी में जब ये आ जाता है GroMax remind you, that means everything is working fine. Okay, so now let us see कि भाई अपना ions कहाँ पर ऐड हुए, right? तो इसे भी you can open it in WinVMD, right? And let us create a representation wherein you can see where are the ions that are added. I am just showing you for representation, just to make sure that things are going in the right direction. Okay, I would create a representation and I would show all the representation. Okay, अभी आपको दिख रहा है यहाँ पर. So ये जो cyan color के हैं, these are chlorine, और ये जो blue color के हैं, these are sodium atoms. So NaCl are added. Now our system is ready, right? We have our protein ligand system in a water molecule which is charge neutralized, right? And our system seems to be ready for the simulation now. Okay, so what do we do next? तो सबसे पहली अपने को चीज जो करनी है, right? ये होने के बाद ये now what happens, my friends, is कि यहाँ पर When you add this water molecule around your protein, these water molecules are also randomly added, and it is entirely possible that whatever water molecules are added, they can form steric clashes with your protein molecules, right? तो ये जो चीज है आपको आपका सिस्टम में स्टेरिक क्लैशेस पहले रिमूव करने होते हैं, and for that you need to perform energy minimization. Okay, so this is where we usually begin the simulation protocol, right? तो सबसे पहले अभी आपको क्या करना पड़ेगा, चार्ज न्यूट्रलाइजेशन हो गया अपना, नाउ वी नीड टू परफॉर्म एनर्जी मिनिमाइजेशन, राइट? तो एनर्जी मिनिमाइजेशन करने के लिए, now let me close this thing. In order to perform energy minimization, you need this MDP file, right? तो हर एक सिमुलेशन करने के लिए उसका स्पेसिफिक MDP फाइल होता है, राइट? एज आई हैव सेड कि जब आप MD सिमुलेशन करते हैं तो देर आर टिपिकल स्टेप्स लाइक मिनिमाइजेशन, हीटिंग, इक्विलिब्रेशन एंड प्रोडक्शन. तो मिनिमाइजेशन प्रोसीजर करने के लिए उसका MDP फाइल लगने वाला है. It is usually this MD or you can say min.mdp. कहाँ पर है यहाँ? Okay, em.mdp. If you open it in any text editor, these are, this is the information that your GroMax need to conduct that simulation. Okay, so it is already there. You can later on kind of check it out कि वही उसमें अगर changes करने होंगे तो. So for every run, now GroMax needs to have its TPR file. ठीक है, तो यहाँ पर अपन GROM-PP यूज करते हैं. In order to perform MD simulation of minimization, उसको energy minimization .tpr फाइल लगने वाला है. और ये TPR फाइल में it contains all the conditions that are there to successfully run your simulation. So these TPR file are for you can say subject, or these are subject to stage specific TPR files. Okay, so just hit enter once you do this thing. Okay, तो em.tpr फाइल तैयार हो गया, right? और अभी what you need to do is to run this md run command. तो यहाँ पर है, ये अगर आपने यहाँ पर पेस्ट कर दिया, GroMax md run कमांड है जिससे आप सिमुलेशन रन कर सकते हैं. Once you copy paste it, hit enter. Now your system has, GroMax has started to perform this minimization simulation. तो अभी minimization में exactly होगा क्या? We are trying to remove the clashes that are there in your protein and water molecule as such. Okay, so it won't take much time. कुछ ही मिनट लगते हैं इसको. Let us wait for time being. Okay, so by the time this minimization is over, is there any doubt that you people want to share or ask? Okay, so let it run. The next step that we need to do is to equilibrate our system, right? तो equilibration ज़्यादातर position restraint अपन करते हैं. That means कि like in our protein ligand system, we don't allow our protein to move, but allow the water molecule to adjust itself around properly. तो इक्विलिब्रेशन में वो चीज आपको करनी होती है. तो उसके लिए आपको पोजीशन रिस्ट्रेंट डालने पड़ेंगे आपके लाइगैंड के ऊपर में भी. एंड फॉर दैट, फर्स्ट थिंग दैट यू नीड टू डू इज टू जनरेट द इंडेक्स फॉर योर लाइगैंड मॉलिक्यूल. ओके, तो आपको अभी इंडेक्सिंग का काम शुरू करना है. Okay, so now it says that the system has converged at the step 906 and everything is fine, right? तो आगे के काम के लिए, for equilibration, you might want to index the ligand. और उसके लिए वी आर गोइंग टू यूज ग्रोमैक्स मेक इंडेक्स कमांड एंड ग्रोमैक्स जेन रिस्टार्ट कमांड. ओके, तो ये काय के लिए होगा? टू जनरेट द पोजिशन रिस्ट्रेंट फॉर द लाइगेंड. ओके, तो ये क्यों करते हैं अपन? बिकॉज ऑल वी वांट इज द सॉल्वेंट एंड आयन मॉलिक्यूल्स टू रिलैक्स अराउंड योर प्रोटीन, and we don't want our ligand to drift away from protein during the equilibration simulation. तो इसके लिए आपको वो ligand का इंडेक्सिंग करना जरूरी है. ओके, तो इंडेक्सिंग के लिए what you need to do is that अपने को सबसे पहले we need to edit the topology file. Okay, okay. So before we do that, अपने को let us directly go ahead. ऐसे भी चल जाए. Okay. आगे का कमांड, we need to index the ligand. तो ये make index GroMax का एक कमांड है. We are going to use it. Just copy and paste it. Hit option. तो यहाँ पर it is now asking for you कि भाई 0 stand for system, right? So on and so forth. We are supposed to give this indexing information 0 and 1 star. Well, let us not go into more details of this thing. For right wing, अपने पास में time कम है, right? Paste it, hit enter, okay, and hit this Q button and again hit enter. So now index.leak file यहाँ पर आपका तैयार हो जाएगा. So that means we have kind of indexed the ligand molecule. That is one of the step that is needed to be done. Okay, उसके बाद में we are going to use this GroMax Gen Register. ठीक है, so I won't go into more details of this right away. आप वो PDF देख सकते हैं कि भाई why is that we are going to do this thing. Now it is asking कि आपको ग्रुप कौन सा सिलेक्ट करना है. ठीक है, so the third group is what we need to index. This is the system information that we have given. Use this third group, hit enter. Okay, and now it has created another file यहाँ पर, position restraint of ligand. Okay, so अभी आगे अपने को क्या करने का है? So this new position restraint file that you have obtained, this position restraint data that you have obtained, ये आपके प्रोटीन के टोपोलॉजी में भी रिफ्लेक्ट होना चाहिए. Therefore, we now need to again you can say update the protein topology file. Okay, तो इसके लिए आपको फिर से क्या करना पड़ेगा? ये topol.top फाइल जो है इसे ओपन करना पड़ेगा. Okay, and in this topol.top file you need to append this information. Okay, जहां पर भी आपका पोजीशन रिस्ट्रेंट का डेटा था, वहां पर करने को somewhere near position restraint for your Fusri.itp. Yeah, need to update this thing. Okay, ठीक है. So all we have to do is to save our work. Now the position restraint is also implemented in our entire system, right? तो ये होने के बाद में we need to again use the GroMax make index command so that we can index again everything into a final index file that will be needed. I will simply copy this command, paste it over here, use this option 1 and pipe, that will index protein which is present on first, and 13, that is your ligand, that is present on 13th position. That is why we are using 1 pipe 13. Paste it and hit enter. Again hit Q to end this command. And now you can see a new file called as index.mdx is created. Right. So now we are done for we are ready for equilibration now. Okay. तो अभी आप आगे के जो सिमुलेशंस है, you can see it over here. आगे के जो सिमुलेशंस है आप दो तरीके से कर सकते हैं. Either you can perform these things manually, right? जिसमें आप पहले आप इक्विलिब्रेशन करेंगे, NVT equilibration करेंगे for 100 picosecond. It would take on my PC, this simple PC, 10 to 20 minutes. After that, you would perform NPT equilibration, which would again take 10 to 20 minutes. And then you would perform production run on 1 nanosecond, that would take 2 to 3 hours. तो आपको हर एक स्टेप के लिए आपको रुकना पड़ेगा. We don't have that much of time really. And उसके लिए what I have done is that we usually do automation. Okay, तो automation के लिए what we have done is that we have created this another script called as pipeline. Okay, तो आपको करना क्या है? All you need to do is to source this pipeline. ये जो फाइल है, right? इसको सिर्फ आपको यहाँ पर कॉल करना है. Just type pipeline.bat and hit enter. And now you can see it has begin the simulation of 5000 steps for NVT. अभी ये क्या कर रहा है आपका? NVT simulation कर रहा है. That means it is performing equilibration in keeping number of atoms constant, volume constant, and temperature constant. Then it would equilibrate your system, keeping number of atom constant, keeping the temperature constant, and keeping the pressure constant. And then it would perform the production run, जहां पर आप आपका सिमुलेशन का फाइनल रिजल्ट्स देख सकते हैं. ठीक है. So this would take 2 to 3 hours, my friends. तो अपने पास में उतना टाइम नहीं है. I have already run this simulation. I will show you the output now. I hope it will be okay with you people, right? तो जब ये आपका ऑटोमेशन खत्म हो जाएगा, it will kind of show you something like this. All simulations completed successfully. That is an indication कि सब कुछ खत्म हो चुका है, and now you can analyze your data. It will show something like this. All simulation completed successfully, right? तो okay. So we are done with simulations now. देखो कितना आसान था. Within half an hour, I was kind of explaining you, जिसकी वजह से इतना टाइम मुझे लग गया है. Otherwise ये कमांड कॉपी पेस्ट करने को कुछ टाइम नहीं लगने वाला, right? And you can easily train your students with this thing. Okay, so I will stop this thing. ये स्टेज पर आपको रन करने देना है आपका सिस्टम for 2 to 3 hours at least. And after you get that message कि all simulation completed successfully, then you can close it. Now I would assume कि अपने simulation complete हो चुके हैं. I will close this thing, and now let us see how our results are. ये मैंने पहले ही कल करके देखा था. Okay, so now let us load the simulations. Okay. Okay, we need to load the Grow file first. We are using VMD. VMD भी एक आपका क्या कह सकते हैं freely available software है. And now we would load data into molecule. I would browse the XTC file, that is generally the GroMax trajectory file hota hai. Okay. It would be with name some MD. Just put this open. ठीक है. Desktop. Now you can see there are 102 frames, this being only 100, I mean 1 nanosecond simulation. अभी आपको बता देता हूँ कि maybe would create a kind of representation that would help you visualize your results properly. And ये जो water molecules है ये सब हटा देते हैं by creating only a protein representation, right? And let us create another representation. Let us select the ligand. अपने protein, इसका नाम अपने को मालूम था कि ligand है. Residue name we are selecting, ligand, and we would create another representation. Okay, so now let us change the drawing style so that you can see the protein in ribbons and your maybe ligand in CPK. Right? Here is your protein, here is your ligand. And let us play the trajectory. As you can see, the ligand is moving inside your protein, as well as your protein is also showing dynamics. You can later on analyze it. Plot the RMSD, RMSF, perform free energy estimation, so on and so forth. Okay, so this is all about, my friends, and I think I would stop now and I would like to take some questions. Okay, so okay, before I take the questions, I would like to address this question now. Okay, like it's okay for teaching. मैंने अभी आपको ये तो बता दिया है कि स्टूडेंट को कैसे ट्रेन करना है. काफी आसान था. कमांड कॉपी पेस्ट करना है, इंस्टॉल करना है, यू हैव एवरीथिंग. बट हाउ डू आई स्केल इट फॉर पब्लिकेशन? ये तो 1 नैनोसेकंड का था. अभी 100 नैनोसेकंड का सिमुलेशन अगर करना होगा तो मेरे पास कंप्यूटर नहीं है. Yeah, I don't have access to the supercomputers. My institute don't have supercomputer. तो उसके लिए there are a few options available. So you can use facilities at National Supercomputing Mission. So using this initiative, Indian government is providing you access to the supercomputers that are there in CDAC. Through CDAC, you can have access to supercomputers that are there in ISERs and IITs. तो CDAC का खुद का एक बायोइन्फॉर्मेटिक्स रिसोर्स एंड एप्लीकेशन फैसिलिटी है, वी कॉल इट एज बार्फ. आप वहां पर भी एक लॉगिन के लिए रिक्वेस्ट करके यू कैन हैव योर ओन अकाउंट. CDAC is also recently providing ICE cloud platform. ये cloud आप खुद के PC के ऊपर न करते हुए you can perform these MD simulations on your cloud platform. So you don't have to end up buying a cluster or buying an expensive PC. Right? तो ये एक ऑप्शन है. अगर आपको CDAC का या फिर ये नेशनल चीजें यूज नहीं करनी है एंड यू वांट टू बी इंडिपेंडेंट, देन यू कैन यूज गूगल कोलैब. You can use Microsoft Azure or you can use Amazon AWS. ये सारे cloud platforms है जिसके ऊपर में आप आपका GroMax install करके you can use it. So this would eventually, it kind of enables you to आपको कोई बड़ा पीसी खरीदने की जरूरत नहीं है. ये सारे प्लेटफॉर्म के ऊपर में कुछ जगह खरीद लीजिए and you are ready to run your simulation. ठीक है, तो this would kind of remove another barrier. ठीक है. So time has come to thank you. So I have a few requests for you guys. Try using the software in your teaching. Try implementing it in your research article. So let other know if you like this software and let us know if the thing is otherwise. Okay. Please write a review on Source4 platform so that it will enable trusting usage of this software in the scientific community. And I would be more than happy to engage workshops and FDP. Feel free to contact me on this number or on this email ID. Yes, Madan. Now I am ready to take it.
Yeah, thank you so much, sir. Actually, I was looking for this since long to learn this. Actually, unfortunately, got you, and thank you so much, sir. I am purchasing one server from one of the project which ANRF recently have granted this early career grant. So how much space is needed for this? Actually, I am in the process of purchase. So the configuration I was searching, actually, I am doing for it DFT. Actually, I do DFT.
Okay.
So along with that, it will be good actually if I could start this also. So what space is needed for this?
Dekho, the thing is that if you have a good grant, okay, and if you can spare at least 5 lakhs or so.
Yeah, I have for this like around 12 lakh.
12 lakh, it is really very good, right?
Yeah.
Then you should go for a Linux machine when it comes to perform MT simulation. तो यहाँ पर स्पेस का ज्यादा कुछ इम्पोर्टेंस नहीं होता है, आपको कंप्यूटेशनल पावर ज्यादा स्ट्रॉन्ग चाहिए, राइट?
Okay.
ट्राई टू गेट ग्राफिक्स कार्ड जितना बड़ा हो सके उतना बड़ा.
Okay.
NVIDIA का लेंगे तो बहुत अच्छा है. You can use it with both GroMax as well as NVIDIA.
Okay. Okay.
So make sure that उसके बाद में कूड़ा वगैरह आप खुद इंस्टॉल कर सकते.
Okay. The problem with the processor only we have to go actually.
Yes. When it comes to DFT as well as MD simulation, processor आपका strong चाहिए.
Yeah. For DFT, I was looking for this Threadripper only.
Yes.
Okay, sir. Thank you.
अगर आपको ये भी नहीं करना है, I mean, you are fortunate that you are having a grant to purchase a PC.
Yeah.
Right. तो
Yes,
आप cloud platform पर भी ये चीजें कर सकते हैं. So
okay.
अगर
So
आपके
I
पास
tried this that one computing mission. So actually we use this.
Hello?
हाँ जी सर.
हेलो.
Yes.
हेलो.
Thank you, sir.
Yes.
Thank you so much.
सर मुझे एक विश्राम सर मुझे एक पूछना था आपसे कि DFT के लिए कोई ऐसी फ्री फैसिलिटी है गवर्नमेंट की?
DFT तो I don't do DFT in my life.
Sir,
No, but still I
can
can
I
help.
interrupt?
Yes, again.
So the same thing actually, you can use this CDAC one National Computing Mission. They can provide space for that.
हाँ सर कोई है क्या ऐसी कि DFT हम जाके हमारे घर पे भी बैठ के लॉगिन थ्रू हम कर सकते हैं DFT की फैसिलिटी?
DFT के सॉफ्टवेयर खरीदने पड़ेंगे. हेलो. यस.
नहीं सर वो सॉफ्टवेयर नहीं, जैसे आपने बताया ना, आपने जो CDAC का जो एग्जांपल दिया वैसे कुछ फैसिलिटी है क्या?
मे बी आप वो सॉफ्टवेयर भी वहां पर अवेलेबल है, आप उनके साथ में एक बार कांटेक्ट कर लीजिए. CDAC में. यस, यू आर ऑडिबल.
हेलो सर, कोई आपने अभी जो एग्जांपल बताया ना CDAC का, वैसा कुछ है क्या DFT के बारे में? वैसा कुछ फ्री ऑफ
अभी उनके पास में ऑप्शन है. यू माइट वांट टू गो अहेड विद दैट. अनिल जी.
Sir, actually CDAC only provide for DFT also. Anil sir. You have option with CDAC only. I am doing this with CDAC only. You can do with National Computing Mission. So there is a facility. Just you need to have a software, as sir said.
Okay.
Thank you, sir.
So our labs, Bioinformatics Center, my lab main objective is to bring this software to every computational chemistry lab, चाहे जितना भी छोटा हो. अगर आपके पास में लैपटॉप भी होगा, if these softwares kind of enable you to get you started up and running, शुरू भी कर सकते हो, तो that is more than enough for us. It will give you a kind of confidence. It will give your student a kind of confidence to yes कि मुझे जमता है ये, मैं कर सकता हूँ. And then once you have that confidence, then it is the next step to deal with the bigger issues कि भाई इसको अभी longer time के लिए कैसे क्या scale up करें, वो बात कीजिए, right? पर पहले सीख तो लेंगे बच्चे, right? ठीक है. तो आज के प्रेजेंटेशन में आई हैव ओनली एंडेड अप विथ generating the data. अभी नेक्स्ट प्रेजेंटेशन में which I have on maybe third I suppose, we would be dealing with कि भाई ये डेटा एनालाइज कैसे करें. उसके लिए भी in my lab we have developed our own softwares. So I would demonstrate those things to you later. Okay, any specific questions, my friends?
हेलो
हेलो
हेलो सर, आपके पास डीएफटी की है के पास?
Yes, we have at Bioinformatics Center. We do have.
सपोज मैंने आपको कुछ स्ट्रक्चर भेजे तो आप कर देंगे वैसे इन कोलैबोरेशन?
जैसे मैंने बताया कि डीएफटी इज नॉट माय डोमेन. डीएफटी अलग चीज होती है, एमडी सिमुलेशंस अलग है. एमडी सिमुलेशंस कम्स अंडर मॉलिक्यूलर मैकेनिक्स, डीएफटी कम्स अंडर क्यूएम. क्यूएम करने वाले अलग लोग हैं एट बायोइन्फॉर्मेटिक्स सेंटर. आई कैन connect you with those guys. No issues with that. Okay. तो अगर आपको एमडी सिमुलेशंस का कुछ होगा तो यू कैन आस्क राइट अवे. Okay, I have stopped my sharing now. Okay, I am open for questions. I think I have bombarded with you with lot of commands. So देखो, maybe someone was asking where is your YouTube channel? Just a minute, I will share my screen. Okay, so there is my own channel called as Scribbles in Bioinformatics. Okay. तो यहाँ पर there are playlists that I have made. Okay, and there are different playlists, like if you want to जो आज अपन ने किया, all the videos that are there are in this playlist, ProMax on Windows, right? If you want to use NAMD, there are 6 videos over here. जिसमें आप NAMD use कर सकते हैं. You want to calculate binding free energy. In our next video, we would be talking with this new software that we have developed in our lab, GroWin Pursuit. It is kind of ये आज अपन ने सब commands through command line किया था. And it is really very inconvenient in the beginning to master the command line. So इसके लिए we have developed a kind of our own interface of ProMax. So we would be dealing with that thing in our next lecture or in our next topic. ठीक है, इससे भी आसान करके. ठीक है. So the name of channel is Scribbles in Bioinformatics. I would request you to look into the playlist, the videos that are there in different playlists, right? सिर्फ MD simulation ही नहीं, docking, most of the bioinformatics things that you might need, it's there. right? Okay. Go through this PDF tutorial. It will help you a lot as well. Okay. This is what I have for time being.
If there is any more query, should I go for vote of thanks? So, myself, Dr. Alok Kumar Verma, on the behalf of MMTTC, Sant Garge Baba Amravati University, Amravati, is thankful to Dr. Rohan Mishram, Assistant Professor, Savitri Bai Phule University, Pune. Dr. Rohan expertly introduced need of MD stimulation to all the participants. The session was highly interactive one. He, through his presentation, made the tedious task look very easy. He showed the participants how molecular dynamics stimulation can be done using GroMax. I am sure many of us may have been encouraged to work on the GroMax for molecular simulation. I again thank to the resource person, Dr. Rohan, for such a nice presentation. We will all be benefited from his presentation. On behalf of all participants, thank to MMTC, Baba Amravati University for providing such a nice opportunity to all the participants. Thank you, sir, for such nice talk.
Okay. I would like to thank Santagaraja Baba Amravati University to give me an opportunity and this software. If Madam allows, then I think I am standing right in the middle of 90 hungry people and their lunch, I suppose.
But thank you so much, sir. I could see so many messages and very informative sessions, and it was really wonderful session. Probably it needs more time. I think it needs an hands-on kind of a thing. So in order to understand all these things. But at least I thought from your lecture, participants they have got some idea about doing molecular docking and MD simulation. So I hope they will contact you and they will be benefited from this lecture, from this session. Thank you so much, sir. Thank you for giving your valuable time. Thank you. So we'll meet.
Should we take break, ma'am?
Yeah.
Thank you.
Okay.
Yes, sir.
Good
Hello.
afternoon. If you could
Good
permit
afternoon. Good afternoon. Yes, no problem. No problem. Please.
So, friends, good afternoon once again. As we have completed the two sessions, now we are moving for today's two another sessions. So, first, integration of technology in our higher education, not only the higher education, but the entire education system now become a very integral part of our teaching, learning, and the research and the entire ecosystem. And therefore, the technology that is the 21st century has been called as the data century. And this technology, that is the integration of AI, artificial intelligence, is very much important now. You know that the couple of thinkers or the policymakers stating that AI will replace us. So such kind of things are also there. If you could not adopt this AI in our perspective, definitely we will replace. So friends, in this session, we will going to discuss the integration of this technology, that is particularly the AI in our teaching, learning, as well as the research. As far as concerned, this session is very much important because one assignment is also there, graded assignment. So that will be explained different tools in this session with the help of one of our renowned expert, Professor Mohammad Atiq sir, who is the senior professor in the Department of Computer Science and the director of this Malvi Mission Teacher Training Center. So first of all, I would like to request Dr. Amita ma'am to please introduce our guest in brief, and then we'll start with the session. Dr. Amita ma'am, please.
A very good afternoon, respected organizers. On behalf of UGC MMTTC, Sant Garge Baba Amravati University, I, Amita Joshi, welcome Professor Mohammad Atiq, who is a professor and HOD of Computer Science and Engineering and Director of UGC MMTTC at Sant Garge Baba Amravati University, Amravati. With over 34 years of academic experience, he is a distinguished researcher specializing in artificial intelligence, soft computing, and medical image processing. He has guided 15 PhD scholars, published 68 research papers, secured multiple patents and copyrights, and authored 2 books. Additionally, he has pursued MBA in HRD and LLB, and NBA NAC assessor and trainer. Professor Atiq has led major research and academic initiatives, delivered over 106 lectures, and continues to promote excellence in research, innovation, and higher education at national and international levels. We welcome you, sir, as a resource person, and we assure you that we will imbibe all the AI techniques learned from you in this session in our teaching, learning, as well as research process. Thank you, sir.
Thank you, Amita madam. A very good afternoon to one and all present here, and I hope I am loud and clear.
Yes, sir.
Okay, thank you, ma'am. Thanks for the confirmation. Good to see you after a long time.
Same here, sir.
Thank you. Thanks for the opportunity. And as far as this session is concerned, we will touch upon one important aspect of teaching, learning, and research. That is the integration of AI into, you can say, our academic landscape. Right. Having said this, as far as this technology is concerned, most of you must be having some apprehension with regard to this, that is whether we should adopt it or we should not, right? So that is a $1 billion question. But as far as I am concerned, you may call me a little bit biased, okay, when I advocate the use of AI for teaching, learning, and research. But at the same time, all of you will agree that there are many things, there are many routine jobs we carry out. Say, for example, whenever you go for preparation of your session or a talk, okay, it is needed to prepare a PowerPoint presentation. Okay, and all of you know that is preparing a PowerPoint presentation itself require a lot of effort, time, and energy, right? So there is nothing wrong in using AI for such routine tasks. And definitely, I am also very conservative about using it for research, but up to some extent, as I said, that is just like PowerPoint presentation, maybe formatting your paper or checking spelling or grammars. So for all those routine activity, wherever the usage is permissible, there is nothing wrong in using it, right? And just like any other technology, this technology again has got some gray areas. Okay, so we'll touch upon all those aspects. Okay, as far as this session is concerned, we'll have two components of it. Okay, the first part we will just try to build some theoretical foundation, and second part we will have some hands-on on some of these tools, right? So having said this, just allow me to share my presentation with you, right? This topic may be a little bit odd, okay, because right now you are undergoing a course in chemistry, okay, but basically we are all teachers, and as far as the NEP 2020 says, it has been mandated, right? It has been mandated by UGC to have at least one session on some of the aspect of NEP 2020. So integration of, you can say, AI or integration of ICT into teaching learning is one of the important component of NEP 2020. So on those lines, we will definitely have some discussion. Okay, I will be happy to take your arguments as well. Some of you may agree or disagree the usage of AI. Okay, so we'll touch upon all those areas. Okay, but at the same time, we need to upskill and reskill yourself, right? So that is equally important, right? So having said this, this is what is there on the agenda. That is, I will start from scratch. I presume that, okay, you know very little about this technology. So we'll start from scratch. That is, we'll just have a look at how this technology has evolved. Okay, what is it all about, and where we are as on date. Okay, and what are the limitations in terms of usage and so on. So if you look at the definition of AI, okay, if you look at the definition of AI, it is nothing but the study of mechanism that think and act like human. That is, in short, what exactly we are trying to do is we are trying to mimic the human behavior. Now, what is so special about human behavior? We as a human being, we have got some peculiar characteristics present in ourselves. Say, for example, we are capable of learning. Second important attribute is we are capable of reasoning, okay, then comprehending certain things. And let's say when you have been trained in a particular area, okay, you are capable of applying that knowledge into altogether new or challenging situation. Okay, so let's say we are in a position to incorporate these four attributes into a machine, we may call that machine as an artificially intelligent machine, right? Now, if you simply look around, you will find there are hundreds of gadgets you are using, okay, wherein this technology is being extensively used. Okay, say for example, look at your mobile phone, or maybe the washing machine, or maybe the air conditioner installed in your office or home. Okay, so there are numerous examples, right? What all these is all about, right? That is what we will just try to understand in the first half of the session. Okay, but before directly jumping into those areas, okay, let us have a dig at how this technology has evolved, right, and where we are at present. Okay, so this slide will give an idea, that is how it has started. Now, most of us, we feel that this technology is new. Okay, but if you look at the historical perspective of this technology, okay, you will be surprised to note that this term was very first time tossed by John McCarthy way back in 1956. Okay, and the event was Dartmouth AI workshop, wherein this fellow, along with few of his colleagues, he has come up with this idea of AI. Okay, and that was the first occasion when this particular term was coined in the research or academic circle. Okay, but 1956 and we are into 2026, so you can very well compute, okay, how many years have passed. Okay, but this technology was there, but it was applicable to only few areas, few specialized areas. There were limitations in terms of computing resources and so on. But as on date, if you simply look around, irrespective of your profession, all of you agree that is we need to have some AI skills. Okay, so those days are not far away, wherein if you are not AI literate, people will call you as illiterate. Right, I'm making some bold statement, but you need to, you may have your own, you can say, view about that, but definitely we need to upskill and reskill ourselves in this particular area. So if you look at the historical perspective, the early AI systems were called as traditional AI systems. Okay. Okay, then we moved into what they call it as the machine learning, then deep learning, and right now we are into generative AI. Okay, wherein now everything is becoming tool based. Everything is becoming tool based. So even if you don't have any computing background, okay, you can very well, rather you can quite easily use all these tools for your activity, right? Whatever daily routine job you are performing, for all those activities, you can definitely use all these tools. Right. So let us begin with what is traditional AI. So traditional AI, the idea was giving the computers the ability to do tasks that traditionally only humans have been able to do. Okay. I'll give you some typical example which will clear your ideas. These systems were basically built using what they call it as, you can say, interacting with the human expert working in that domain. Okay, let's say you want to build a expert system, and these systems were referred to as expert system initially. Okay, so if you want to build a system for, let's say, medical diagnosis. Okay, so you may approach a medical practitioner, ask him or her a number of questions, and based on the responses, you can very well frame certain if-then-else rules, right? And those if-then-else rules are basically fed into a machine or a computer or other programmed, explicitly programmed, okay, to achieve the automation of that activity, right? The typical examples are like, say, playing chess, or maybe homeopathy expert, or if you look around, like, say, washing machine or the microwave oven or the air conditioner and so on, right? But there were limitations with regard to these traditional AI systems. What are all those limitations? The limitations were like, say, they were time intensive. Why I'm saying time intensive? Large rule bases. Okay, if you are dealing with a very complex problem, okay, you need to frame hundreds of rules, rather thousands of rules. Okay, and for searching all those thousand rules, okay, definitely it will take little bit more time. Okay, so time intensive due to the size of the you can say the rule base. Then the another issue was it doesn't generalize to new situation. Now, what do I mean by new situation? Say, for example, if you have designed a homeopath medical diagnosis system, okay, and you are facing altogether COVID-19 kind of a scenario. So in that case, what will happen? It will come up with the answer. I'm sorry. Okay, it's not possible for me to diagnose this particular disease, right? Because those symptoms are not matching. Okay, so you may have a no match kind of a scenario. Okay, and that led to what they call it as the doesn't generalize to new situation. Okay, another important aspect, as I have referred to, that is whenever you develop these system, okay, you need to interact with the human expert working in that domain, right? So this is the you can say the this is little bit related with human psyche. That is, we know more than we tell. Okay, that is also what they call it as the Ferrari's paradox, right? So we know many things, but we tell many times less. Okay, and if you are dealing with a very complex problem, it is not possible to devise rule for every possibility, right? So that may lead to what they call it as the combinatorial explosion. Okay, if you are dealing with only 3 parameters, just like in case of washing machine, you can very well devise around 27-30 odd rule, and you may automate that job. But let's say if you are dealing with a very complex problem, Priyanka
बोलो
Singh.
नहीं 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 स्कूल जाना है.
Okay, so it may lead to combinatorial explosion, right? So that makes it a bit difficult to address all the issues associated with that specific problem domain. So as to address all these issues, we moved into an era of what they call it as the machine learning. Now, what is machine learning? That is the second block. If you look at the slide number 2, okay, what is it? It is, instead of explicitly programming and telling the computer what to do, what we are doing in this case, in case of machine learning, we are exposing the computer with lots of input output data pair. Okay, and we are using some or the other statistical technique to learn the relationship between the input and the output. Okay, so here we are not explicitly programming, just like we used to do in case of traditional AI or classical AI, okay, wherein we used to frame if then else rules. But now we are not doing so. Rather, we are feeding the machine with some raw data, asking it to learn from that data, okay, by using some or the other statistical techniques. Now, under statistical techniques, we have very many possibilities. The possibilities are like, say, linear regression, maybe logistic regression, classification and regression, support vector machine, random forest, gradient descent, and last but not the least is the neural network. Okay, we'll have some detailed discussion on neural networks, right, in the slides to come. But these are the various possibilities, right? And typically, if you look at the mathematics involved in this gradient descent, okay, you will be surprised to note that it was proposed way back in 1880. So the roots of this technology are quite deep, I should say, right? It is not, it has not happened within 5 to 10 years or 20 odd years, but the roots are quite deep. Okay. Anyway, so we may use any of these technique to learn the relationship between the input and the output. But there were issues associated with machine learning as well. What are all those issues associated with machine learning? The number one was, that is, we have to have structured data. Okay, as I have told you, that is, we need to have some data set, okay, which are nothing but numbers. And to be very precise, numbers between 0 and 1. Right. So we have to have structured data, or the data which can be numeralized. Okay. Now, once again, we consider the same example of medical diagnosis. Okay, wherein certain parameter values not necessarily will be in the form of numbers. Okay, maybe text or something like that. Some description is there. So what you need to do is you need to convert those text into numbers. Okay, so we have to have specifically numbers between 0 and 1. Let's say if you are getting some 3-digit or 2-digit number or 4-digit number, what you can do, you can divide it by some constant like, say, 10, 100 or 1000. Okay, and you can very well convert it into, you can say, number between 0 and 1. So we need to we have to have numeralized value, right? So we need to perform some sort of pre-processing. So this is the fair overhead associated with machine learning. Okay, that is the very issue was, we need to have structured data in case of machine learning. If you are having a structured data with regard to any problem domain, you can very well use it for machine learning activity. Okay, but there were issues. Okay, and as I have told you, one of the important some statistical technique is the neural network. Okay, now we'll just try to have some detailed discussion on neural networks. So basically, neural networks are biologically inspired artificial neural network. Okay, so this is the structure of a biological neuron. Okay, in our brain we have this kind of neuron which are present in billions. Okay, and they are connected with each other. Okay, what is it? As a matter of fact, it takes its input from the dendrites. Okay, then some processing activities carried out in the cell body, and when whatever is the output, it goes through the axon to the another neuron in the network. Okay, so this is a simple biological neuron. Now, on the similar line, we have an artificial neuron. What is it? This is just like, say, X1 and X2. These are the two neurons. Okay, and with each neuron, we are having a weight associated with it. Now, what for this weight is used? This weight is basically responsible for the learning capabilities of neural networks, right? So if you are using ChatGPT or any other, you can say, large language model, on the bottom line, there are neural networks. Okay, there are neural networks. Okay, and the learning capabilities of neural networks are due to these weights. Okay, they are that is also referred to as what they call it as the synaptic strength. Okay, in case of a biological neuron, you have some synaptic strength. Okay, and that is basically depicted by W in our case, that is in artificial neural network. So, and then what do? We compute the output. Typically, this y is equal to summation of x1 into w1 plus x2 into w2. Okay, so this is a single neuron. Okay, but the things are not that simple, right? Just like your human brain is very complex, on the similar line, these artificial neural networks are again very complex. Okay, this is a single layer perceptron. But let's say if you go on adding the layers, it will look like something like this. Okay, so wherein you have around, say, you can say x0 to xn as the input. Okay, okay, so whatever is the application area you are dealing with, let's say you are dealing with a handwritten numeral recognition, and you are extracting around 40 odd features. So this will be around, say, x0 to x39 input neuron. Okay, through which you will feed the data to the neural network. Okay, then as I have told you that with each x0 there is a w0 associated. So x0 w0, x1 w1, and up to x39 w39. What do we do? Then we sum up these things, okay, in the cell body. Okay, so summation of x0 into w0, x1 into w1 plus and so on, xn into wn, I and j equals to 1 to n, right? And then what happens? Whatever is the output, it goes through some non-linearity. That is what they call it as the activation function. We'll not go into the details because it is beyond the scope of this session. Okay, if I go on talking on those lines, okay, it will become a little bit difficult. But at the same time, we need to know certain basic aspects. Okay, that's why I'm touching upon all these things. Okay, and then we get some or the other output. So in the training, what do we do? Or in the training or learning, what do we do? We provide the input and we start with some random weight. Okay, as I have told you, all these values, whether it is x0 or w0, it is between 0 and 1. So let us start with a, you can say mean, okay, 0.5 of all the weights value, okay, and then go for the computation. Okay, so you will get some or the other output. Let's say your desired output is 1.0 and the actual output you are getting from the network is 0.85. Okay, so meaning thereby there is an error of 1 minus 0.85, that is 0.15 is the error. Now, what we can do, we cannot change or alter the input. Okay, the only thing in our hand is to change the value of these weights. Again, there are two possibilities. Either you can make it, rather you can increase it, or you may decrease it. Okay, let us try to increase it. So from making 0.5, let us make it 0.6 all, and again observe the result. Let's say in this case, if you are getting output as 0.9, earlier it was 0.85, now you are getting 0.9, meaning thereby you are in the right direction. So we go on increasing the value of weight till the value of this error becomes 0 ideally. And at that point of time, we will stop training. We declare that the training is over. Okay. So during training, what do? We try to normalize the weight. Okay. And then let's say if you expose this network to any other thing, any new pattern. Okay. Once the training is over, okay, you can expose it to any new pattern. What it will do, it will not come up with anything like, say, no match kind of a scenario. It will give you some or the other answer. That answer, I am not saying all the time you will get as the correct answer. Okay, it may be right or wrong, but you will get some or the other answer. So having said this, this is how the training basically takes place. Okay, in a very broader term. Okay, definitely there are many things involved. There is what they call it as the local minima, global minima. There are very many things. Okay, how to come out of that local minima? Again, there are you can say momentum term. Some constant is supposed to be added, and so many things. But on a broader perspective, this is how the training basically takes place. Again, supervised training is there, unsupervised training is there, semi-supervised training is there. There are very many options. One more category is there, what they call it as the reinforcement training. Okay, so reinforcement training is something which is basically trial and error. Okay, it is very similar to when a small kid learn how to walk. Okay, that is what is reinforced. Try till you achieve the desired result. Okay, trial and error, and that reinforcement is basically used by all the large language models. Right. So I think this is enough as far as the training aspects are concerned. Okay, then certain terminologies are there. I'll not go into the details, but we have seen that is how to update the weight. And then when you cycle through from this input to output, this is called as one epoch or one iteration. Okay, this is called as one epoch or one iteration. Okay, so this way you may set 500 epoch, 1000 epoch, okay, 5000 epoch, and so on. Okay, so this is how the training normally takes place. Then if you just recall slide number 2, the third area is what they call it as the deep learning. Okay, again machine learning, then we have deep learning. Okay, in case of deep learning or DL architecture, on the bottom line it is basically a neural network. Okay, the only difference is that you are having hundreds of layers, and that's why it is referred to as deep learning. Okay, so deep learning architecture there's nothing new. Okay, you have number of layers are quite large, and that is why they are referred to as deep learning neural networks. Right now, what are the limitation in terms of deep learning? Now, the best example, okay, you are using in your day-to-day life. Okay, whenever you switch on your mobile phone and expose your face, okay, so it will recognize you whether you are an authorized person or not. So what is it exactly? On the bottom line, it is somewhere it is using some algorithm. DL algorithm is running. Okay, so whenever you purchase a new handset, what do? You train it by exposing your face from different angle so that it captures certain features and stores those features into the algorithm. Okay, and next time whenever you switch on your mobile, what it does, it compares those features with the stored features. Okay, if there is a match found, it will say the legitimate person, the mobile will unlock. Otherwise it will ask you, sorry, unrecognized face, or some error message will pop up. Right now, in case of deep learning, in terms of input output modalities, it is structured or unstructured. If you recall, in case of machine learning, we have to have only structured data, only numbers. But in case of deep learning, you can give either structured or unstructured. What do I mean by unstructured? Like your image or your photo, right? So that can be given as input. What about the output modality? Once again, structured data. Okay, either unlocking a mobile phone. Okay, so either it will recognize you or it will say, sorry, okay, I am unable to recognize you as a legitimate person, and it will not unlock. Okay, so deep learning in terms of input, structured and unstructured, output is structured data only. Still, we are in structured data only, right? Now, we just try to recall the fourth block. It was generative AI, if I am not wrong. Okay, this is the era wherein we are at present, right? Generative AI. What is generative AI? If you look at the hierarchies, at the top you have AI, then you have machine learning, then you have deep learning, and the innermost layer is the generative AI. So it is the small subset of AI. But if you look at the potential of this generative AI, you will be surprised to note that these generative AI tools are capable of creating new original content. Okay, so new, I underline the word new. Okay, what do I mean by new? New meaning thereby which didn't exist before, completely new. Okay, let's say you want to write a research article. Okay, or you want to create an image or rather painting, completely new painting. You can do quite easily with these tools. So creativity, okay, creating new original content such as images, text, or even composing a music is possible by means of these tools. And that is the real power. I say power as well as I will put a slash. That is the real threat for all of us. I hope all of you will agree with me, right? That is the real issue associated with this technology. So what it is doing exactly, it is challenging your creativity, innovation, right? Which is typically associated with human intelligence. And that is the real glitch, right? So one has to understand this from those point of view. And definitely, most of us will disagree that we should not allow the usage of this technology, okay, for these purposes. Anyway, having said this, in terms of generative AI, okay, what about the input? Now the input is completely unstructured. So you can give text or image as the input. What about output? Again unstructured. Earlier, in case of deep learning, it was structured only. That is, unlocking your mobile, say yes or no, 0 or 1. Okay. But now, what is happening is you are having numerous possibilities. Why I'm saying numerous possibilities? Because you can write a prompt and ask it to generate an image. It will do it for you. So text to image is possible. The vice versa is again possible. You give an image, ask it to describe that image. It will do it for you. Right? You may write a prompt that too in your native language, like say English, and ask it to compose a music for a particular song. Quite easy, right? Then text to text, what we are using right now, okay, which is the most common aspect of this generative AI. Okay, now under this category, you have numerous possibilities. Okay, numerous possibilities are there. But the real power of this Gen AI is its multi-modal functionality. Multi-modal functionality meaning thereby you can give text plus image as the input and you may get text plus image as the output. So multi-modal is not merely, you can say the text or image, it may be a video or so on and so on, right? So multi-modal in terms of those aspects, right? And that is the real power of this Gen AI tool, right? Now, how it is different from your traditional AI? Okay, now we'll just try to sum up whatever we have discussed till now. Okay, if you look at traditional AI, it is typically used for, or the primary goal was recognition and classification. It will recognize, okay, some disease or classify something, let's say handwritten numeral 0 to 9. But generative AI is, yes. Sorry, I'm really sorry, sir. Please mute yourself.
म्हणजे 15 जूनचा होता तो पोस्टपोन झाला आता. कोणते जून पण होता.
Hello. I request the participant to please mute themselves. Okay, so creation and synthesis. We will have a look at these aspects as well. Okay, that is what this creativity is all about and how to synthesize the input and so on. Right. Now, let us move to the output type. Okay, you can create new original content and that is the real threat, right? So you can create completely new original content which didn't exist before. Okay, then what about the data usage? If you look at the data usage, okay, you will be surprised to note that in case of traditional AI, it only analyzes the data, whereas these Gen AI tools, they are capable of learning from the data. Okay, so the learning takes place from the data itself. And what about the interaction? Interaction is, in case of traditional AI, is only one way, but whereas in case of generative AI, it is interactive and conversational. Okay, so you may interact with them as if some person is sitting on the other side and that fellow is obeying your instruction and accordingly crafting answers for you, right? So that is the real power of this generative AI. Now, having said this, what are the core models of this generative AI? The core models are, or rather core components of this generative AI are many, but the very first one is the foundation model, which is typically used, you can say, as the base model. Okay, as the base model. Okay, and they are basically trained on the massive diverse data sets, right? That is the foundation model. Now, if you look at the most common, you can say the, I'm sorry, I request the participant to mute themselves so that I can continue. If you have any specific query, you can fire it now. Hello. All right. So where were we? We were talking about the large language model. So LLM, all these LLMs are capable of predicting the next word, right? Now, I hope all of you will appreciate this. That is, let's say, for example, if you give all that glitters as the input, they are capable of coming up what they call it as is not gold. Okay. Now, there are definitely this sort of response will definitely require some sort of intelligence. Okay, at least on this point, all of you will agree, right? So they are using AI at the bottom end, okay, and these large language model like say ChatGPT or a DeepSeek, anybody, any tool you may assume for that purpose is typically using this kind of a thing, right? The another important core component is the transformers, okay, what they call it as the attention mechanism, okay, which allows the model to understand the relationship between different words or data points. That is the second important thing. Then you need to train and fine tune them, okay. So you may use some foundation knowledge, and then later on you may fine tune it with your own data. That is again possible. We'll have a look at those aspects as well. Then if you look at the life cycle of these, you can say the Gen AI tool, okay, there are 4 phases, okay. The very first one is what they call it as the pre-training, okay, wherein it is exposed to a diverse range of data, okay, and we train it for that purpose. Then we have fine tuning, wherein we refine the performance using some specific use case data, okay. And then we have an inference mechanism, okay. So and last but not the least, how do they improve? They improve by continuous feedback, okay. So there is some feedback mechanism. Based on that, it improves its own performance, okay. So these are the 4 phases of the life cycle of a Gen AI tool. Now, having said this, there are some popular series, okay, like say GPT series, which you must be well acquainted with, which is basically developed by OpenAI. Okay, then we have Dell E and Midjourney, which is having creative AI kind of functionality. Then we do have IBM is again a lead player, but this technology has got certain problems as well. Somewhere I was referring to, there are some gray areas. So one of the gray area is what they call it as the black box. Okay. Now, what do I mean by a black box is basically it whenever you give an input to these tools and it will give you some or the other answer, you never know that is how the tool has arrived at that particular answer. So whatever is happening within the tool, it is not known to anybody. I underline anybody. So, anybody meaning thereby, even if you approach OpenAI people, okay, or the you can say the person involved in development of those tools, they will again come up with the same answer that they don't have any justification. Why? Because the complexity is so high. There are millions of layers and hundreds of layers and millions of neurons. So that makes it a bit difficult to justify how a tool has arrived at a particular answer, right? So that black box, now there are solutions to it. Okay, people are working on that. We have something like what they call it as the transparent AI or the responsible AI. Okay, wherein this add-on functionality is being added to the existing tool. Okay, which will give you the justification how the tool has arrived at a particular answer. And that's why usage of these tools, okay, in critical application is strictly prohibited. Say, for example, if you are using it for medical diagnosis, okay, and let us assume that it is diagnosing some disease which is, as a matter of fact, not there, okay, but the doctor has started treating you on those lines, so you may face problem. Now, again, there are some legal issues as well to whom you will sue, and there are many things. So black box is one of the bottleneck of this technology. Now let's come to our focal point, that is what are the use cases, particularly with regard to education and research. Now you may use these tools for auto generating lesson plan and build application, I tell you, within no time. I'll show you by writing a simple prompt in English, you can build an application. Okay, there are you can say agentic kind of environment provided by Google itself. Okay, so we'll have some hands-on on those lines, but you can do all these things quite easily using these tools. Okay, that is for education. Okay, the use cases are many. For research, again, summarizing or maybe dense research paper, analyzing those papers, again, that is nowadays new normal. You can do it quite easily. But on the, you can say the commercial front, again, there are hundreds of applications. I've listed out only few here. But marketing, healthcare, finance, again, there are many things. Right. Now, how do these LLM basically works? Okay. So these LLM basically works in 2 phases. Oh, so sorry, 5 phases. Okay, rather 5 steps you may call it. Right. The very first step is what they call it as the tokenization. Okay. So in tokenization, what do we do? Let's say you have written a prompt wherein you are having around 50 odd words. So for simplicity, you may assume each word as one token. Okay, many times it is more than that. Okay, for example, say in a simple word, if you are using ES or some apostrophe S, okay, it will be considered as another token, because that token will help the tool to understand the context in which that prompt has been written, right? So the very first step is the tokenization. So whatever is the prompt, okay, whatever you type in English, each word you may consider it as one token for simplicity, but it is many times more than that. Second is the embedding. Once you are done with the tokenization, second step is embedding, converting those words into numbers or numerical vectors, because it is not possible to process those words directly as it is by some computer. Okay, so you need to convert them into vectors. Then we do have a self-attention mechanism. Then the fourth step is what they call it as the predictive capabilities, which I have already demonstrated to you. That is, they are capable of predicting the next word in the sequence. And at the end, what do? They generate the response. Okay, so based on the context, okay, and the intention of the you can say the prompt, accordingly the response will be generated. So these are the 5 steps in which these LLM basically works. Right now, what we are left with? We are left with only one thing, what they call it as the prompt engineering. Now, how to write a precise or you can say accurate prompt? Okay, that is what is left in our hand, right? So prompt engineering talks about all those aspects. Now there are hundreds of prompt engineering techniques. Okay, but we'll not go into the details of all. But just for the sake of example, just try to have a look at maybe 1, 2, or 3. Right. So the very first one is what they call it as the zero-shot prompting. So wherein you don't give any example. Okay. So whenever you are new to these tools, what do you do basically? You ask a straightforward question without giving any example. Okay, and that's why you get more generic response in that case. Okay, you don't get the response which you expect from all these tools. Okay, so many times you get generic responses because you are firing a direct question without giving any example, and that is typically zero-shot prompting. So initial our instruction is always zero-shot prompting, right? But let's say if you give one example, okay, and then ask exactly what you intend to ask. Okay, it is called as one-shot prompting. So it becomes a bit easier for the tool to craft answers for you, right? So one shot, we give one example. There is one more technique, what they call it as few shot, wherein you give more than one example. Okay, so if you give examples, it becomes easier for the tool to understand what is your intention, and accordingly the answers will be crafted for you. Okay, so zero shot, one shot, and then few shot I have already told you. Okay, that makes you can say the job of the tool little bit easier. What is your intention, and accordingly the answers will be crafted for you, right? So few shot I've already told you. Then there is a role prompting. You may assign a role. Okay, you may assign a role of a research assistant, okay, to a particular tool, and say for example you may say, okay, boy, assume you are a vice chancellor of Sant Gadge Baba Amravati University, and then you pose your questions. So okay, so it will become a bit easier for the tool to craft answers for you. Then we may have chain of thought. Okay, there are many things, but I don't think that we have that much amount of time, because we need to go for the demonstration as well, right? So chain of thought, wherein you break down a task into smaller ones and then fed it one by one. Meta prompting, wherein you are asking the tool to generate prompt for you, but this definitely requires more expert level of reasoning and sophisticated responses. Okay, then tree of thought, wherein it will evaluate multiple options, and then it will give you the you can say the most optimal solution for a particular problem, right? Whatever is the problem you are asking. Okay, then prompt chaining is there. Then there are few things which we must know as a user. What are all those things? The things are like, say, there are few parameters which we must be concerned about. So the very first parameter is what they call it as the temperature. So this is our usual complaint. Whenever you interact with these tools, what happens? you feel that you get disappointed that you are not getting the right answer, okay, or the more precise or the accurate answer, because these values of these parameters are not properly set. I'll show you how to do it, okay, in the demonstration. But the very first temperature on those lines is the temperature. Value of this temperature is between 0 and 2. So if you keep it small, okay, you get more generic response. Okay, more generic response. Okay, if you want to increase the creativity, you need to increase the value of this temperature. So if you make it 1.5 or 2.0, you will get more creative responses from the same prompt. Okay, prompt you will not change. Okay, so it is directly proportional to the creativity or randomness, right? So we need to be concerned about this particular parameter, temperature. Second is the max token, okay, which defines the length of the response. You can set it quite easily. Okay, longer token limits result in definitely more detailed answer. Okay, so then you have top P, wherein how many potential word it must use. Okay, you want to craft it for a specific thing. And then frequency penalty and presence penalty. Many times we get redundant responses. Certain things are being repeated again and again. If you want to avoid that, you need to set these frequency penalty and presence penalty appropriately. Now, does this mean that Gen AI is the end of the story? Now, I'll take a pause for a minute, for rather few seconds, okay, to get your response. What do you feel? Whether Gen AI tools are foolproof? There are no issues associated with this Gen AI?
No, sir. I think it is not foolproof.
Yeah. Again, there are lot of I'll say one complete slide is dedicated for that. So it suffers from, again, we have another set of problem. I'm not saying that it is not at all good or bad. Okay, but there are issues associated. Now, what are all those issues? Let us have a look at. All these LLM, why we are considering it? Because this is Gen AI, generative AI, right? Suffers from hallucination. We are all teachers, Okay, whenever you interact with your student, okay, they even if they don't know the answer, they will try to attempt it. On the similar line, these tools, even if they don't know, the tool doesn't know the answer, it will answer a query that it doesn't know. It will give you some or the other answer, maybe many times wrong, right? Then it has got what they call it as the bias. Bias is the another problem. Okay. Then knowledge cutoff. Okay. Somewhere I've coined the term pre-trained. So all these tools are trained up to a specified date. Say, for example, the training took place in December 2025. Okay. Then it will not have what has happened from December 2025 to June 2026. Okay. So you need to ground it. Okay. That I'll again show you that is how that can be done. It can be done quite easily. Okay. So if you are asking question in June 2026, whereas the training took place in December 2025, okay, definitely you will not get the appropriate answer. So knowledge cutoff, how to address it, that again we'll see. It doesn't know about you or your organization. Okay, many times they are not, because they are all general purpose tools, right? ChatGPT is for everybody, right? Okay, so it will never note whether Professor Mohammad Atiq is interacting or Professor Anup Biswas is interacting, right? So, or anybody else is interacting. So that is where you need to train it with your own data. Let's say if you want to use it for your own organization, okay, or for your own personal use, you need to train it with your own data. Okay, so that is another thing. Then training cost of these tools is quite high. That's why none of these tools are available free of cost. Okay, some beta version they provide you, okay, free with some limited functionality, but not all features will be there. Then latency, many times it takes a lot of time, say analyzing, synthesizing, researching, okay, and then coming up with the answer. So that is the another bottleneck. Then all these LLMs has got some token limit. Okay, that is they can handle only 1 million token per chat. Okay, so there are numerous problems associated with this LLM. Now, if you want to address all these issues, okay, what you need to do is you need to go for a solution, what they call it as the RAG. Okay, now I don't know whether we have that much time, right? But in RAG, what do we do? Okay, we retrain it, okay, the pre-trained model with our own data, organizational data, you may say, or your personal data, okay, and then you adapt it for your own usage, okay, then and then you can use it for your own purpose. So somewhere we'll have just some talk on those lines as well. That is how to build your own large language model. But anyway, what RAG is all about: retrieval augmented generation, wherein while using it or while retraining it with your own data, you need to carry out some pre-processing sort of thing on the data. Okay, what they call it as, now there are various things involved. The very first one is the chunking. Okay, let's say you have got a 1 TB of data, one single file, or maybe 50 GB of data. Okay, and you fed it in a single go to the machine. What it will learn from that? Okay, so you need to divide it into small chunks. That may be maybe 500 tokens, and then these tokens, so as to understand the connectivity. Okay, many times we need to go for what they call it as the chunk overlapping. That is 450 to 500, and maybe next chunk 0 to something like that. So these things can be overlapped, and then so as to establish the connectivity, understand certain things. Then embedding is there, okay, which we have already talked about. That is, we need to convert these words into numerical numbers. Then vector database you need to create for your own organization, and then you may apply any of the similarity search, like say Euclidean distance or maybe cosine search and so on. So RAG is the solution to almost all these problems. Almost. Knowledge cutoff will be addressed, bias will be addressed, okay, doesn't know about you or your organization can be addressed, okay, and so on and so forth. And even training cost is addressed, because earlier you are using some pre-trained model. Now you are not retraining it. You are providing some extra training, you may say, with your own data. Okay. Now this is the overall scenario of this RAG, wherein you can say the input query, then the retriever is there, okay, and to support this retriever we have our own you can say the organizational data, okay. So the generator, that's why this bidirectional arrow is there, right? So this is how basically it works, right? This is how basically it works, and there are numerous advantages. Okay, it's not possible to talk on all the lines, but I'll just read it out for just the sake of your information. You may have factual accuracy here, reduced training needs. Okay, why I'm saying reduced training needs? Because you are using some pre-trained model. Then multi-domain retrieval. Okay, that is again possible. Secure use of proprietary data. Your own data, if you give it to some third party, okay, definitely it is vulnerable in terms of privacy and security. So you can address it, okay, because you are doing it on your own. So it is in your custody only. Then it is modular and flexible, and so on, right? So RAG key features, if you look at, okay, it uses a philosophy, what they call it as look up before speaking, right? You have factual foundation, that is domain accuracy can be improved. Okay, then you may deploy it for some real-time uses. That is again possible, and it is very flexible. Okay, having numerous advantages. Then grounding actually we are achieving by adding up our own data. Okay, then transparency and explainability. Okay, because things are almost in your control. Then it is future-proof architecture. Okay, so we are basically decoupling the data retrieval and generation. So there are three small components, okay, which allows the RAG system to evolve with the new models. So you can build new models quite easily with using this RAG architecture, right? So this is the factual AI standard, okay, and this is how this RAG is all about, right? Now, how to use it for research? Now, here definitely some of you may be having some disagreement with me that we should not use it for research. Okay, and there are reasons to it. Okay, so as far as I am concerned, you may use it for only you can say research, wherein you can say the publishing houses or maybe the uh, players involved in the publication, okay, they permit you, okay, for only those purposes you may use it. But not for idea generation, okay, or not for creating one complete article. Because if you are doing so, definitely you are compromising on the ethical front. So we are all teachers, and definitely there are chances when you produce some article using these tools, okay, definitely you will face problem. Okay, so say for example, right now I am from engineering discipline, so we have IEEE mandates wherein it allows the use of these tools only for editing and grammar enhancement, not for anything else. Okay, so one should not use it for anything other than what has been specified in the guidelines. Okay, then for education, definitely you can use it. Okay, I'll show you assessment, you may use it. Okay, you can create question papers. There's nothing wrong in that. But again, it is vulnerable in terms of, you can say, security. Okay, so we'll address all those issues. That is how to, without making any compromises on security, how to deal with these Gen AI tool particularly for assessment. Then teaching learning, you may use it, research quality, and so on. So this is an indicative list. Okay, but we'll have some hands-on on few of these tools, and definitely I'll give you some assignment based on this. Okay, which you are supposed to submit on the Samarth portal. But before that, let's say if you have any specific query, you can fire it. See, the
Thank you,
first
sir,
part we are
for your-
almost done. Yeah, Vishwas, sir, please.
Thank you, sir. Thank you for your nice presentation on AI, the hot topic of AI with today's perspective. So I have a couple of questions here. So first of all, most of the publication houses are now restricting the AI for creating the whole article. That's true. But some of the publications allow the AI for editing and grammar enhancement.
Yes.
Is it also acceptable? Because if you write a paragraph with poor sentence construction and if you rephrase it with AI, then you will get up a new developed paragraph. And with the help of the AI, you can come up with the conclusion part or the introduction part of an article. Is it also acceptable?
Yeah, actually, there are deviation, whatever you are saying. That's why I said you need to refer to the guidelines published by the various publishing houses, because they don't give similar treatment. There is no simple one single code which is applicable to all, right? They have got their own way of dealing with this. But again, why I'm saying that one should not use it for the creation of the entire paper, because whenever you put that paper in the plaque check, what will happen? It will be categorized as what they call it as, sorry, Nilima madam. Okay. So what I was referring to, that is when you put anything, something you have paraphrased, okay, you are pasting it and then you are putting it for the plaque check. So plaque tools are becoming equally competent. They will identify or categorize that particular stuff as the AI generated stuff.
Yes, sir. Here is the second comment that suppose you are copying up well-reputed paper from a well-reputed journal, and if you are replacing it, you will get a new paragraph based on the same theme. And if you go for the plagiarism checking, it will obviously pass the plagiarism checker.
No, it will not. That is what I am saying. That is why I am saying AI generated stuff is basically flagged as AI generated stuff. It's not, some cases it may happen, but not all the cases. Somewhere you will be definitely caught, if I am not wrong, right? Because these AI, see, plaque tools are having this AI functionality built in most of the plaque tools. Maybe Turnitin or Authenticate, if you look at, they have got this AI detector. Okay, built-in AI detector is there. So it will be categorized. I have never experienced it, but I'm damn sure that it will be categorized as a AI generated stuff. Okay, you will not get a go through from the tool. Okay, it will not be in a position to say exactly which tool you have used, but it will categorize it. It is a AI generated stuff. Some tool you must have used. Okay, it will not come up with the exact name of the tool, but definitely you will get a red flag over there. Okay.
Okay. Okay.
That is my
And
take. Okay. Yeah.
okay. And my second question is that, sir, you have in your lecture, you have mentioned that temperature dependent creativity. That means the Gen AI, if the temperature is higher, then the creativity will be higher.
Yes. Creativity or randomness.
Randomness. How creativity is equivalent or directly proportional to randomness? That means the more creative people are more random.
Actually, these are the two terms which are typically tossed there. Okay, and that's why I specifically use both the terms. Okay, they are synonymous terms in AI technology or generative AI specifically. Creativity is more than actually randomness. Okay, randomness may be a subset of creativity, but some of the you can say the literature you will find it is referred to as randomness. Okay, that's why I've used both. That's why I've used both. Yes.
Okay.
I hope I have
Thank
answered you. Okay.
Thank you. Thank you.
Okay. Now let us begin with certain tools, demonstration of some of these tools. I hope there are no more questions from the participant. So let us go ahead with the usage of these tools. Whether you are using some of the tools, just inform me. If yes, say yes or no. Otherwise, I will start with a very basic tool. Okay. And because I need to take into account, okay, there are two types of participants. Some of you must be already using Claude and then anti-gravity and whatnot. Okay, but at the same time, there are few people which are a little bit conservative in their approach. Okay, they may not be using it. So we'll start from a very basic tool called as the Notebook LM. Okay, wherein you may expect some sort of assignment as well. So it is a very, you can say, fantastic tool. Right now, it is available free of cost. Okay, so I hope my entire screen is visible to you. Whether you can see my entire screen?
Yes.
Say yes or no.
Yes, sir.
Okay. Now I am going to my Gmail account. Okay. So just log into your Gmail.
Sir,
Go to the
slide is
We can't see, sir.
Acha, my slide is
Sir,
visible.
only thank you. Yeah, thank you so much.
All right. I will once again share it. Okay. What has happened? Probably I have shared my slides only. Let me share the entire screen. Now probably my entire screen is visible to you.
Yes, sir. Yes, sir.
Okay. So log in with your Gmail account, go to these Google Apps somewhere at the bottom. I may be a little fast, okay, from this point onwards, because there is no rocket science involved as a matter of fact. Okay, some of you must be using this, you may be a little bit well acquainted with all these things. Okay, so this tool, the Notebook LM, it is basically a product of Google. Okay, and it is being used extensively for teaching, learning, research, or you may say for very many things. Even assessment, one may use it. Okay, so this is my Notebook LM. Okay, right now, as I've told you, this is completely free. Okay, so you may let's say you want to create a new notebook. Okay, so you may go ahead with this create new button, or the other way around, you have create new notebook here as well. So click any one of them. Okay, then you will find something like this on your screen. Okay, now let's say if you are using for research, okay, and you have already downloaded few files. Okay, all those research papers can be uploaded using this upload file button. Or let's say if you want to extract some certain things from YouTube or certain website, you may use this option. If it is the stuff is lying on your drive, okay, all those papers you may extract or upload them from this drive option. Or let's say if you have copied certain things and you want to paste it, okay, so you may use this copied text all option, right? Or otherwise, let's say we want to search it on the web. Okay, so let's say AI in you all are from chemistry. Okay, so I'll use analytical chemistry. Okay, this is the topic brought just for the sake of example I am giving. Now you have various things here. Okay, fast search. Then you may go for a deep search. Okay, as per your own need. Yes, sir. Sir, fine. नहीं ठीक ठीक जी so I'm really sorry for that but anyway so this is AI in analytical chemistry. Okay, and then simply hit this blue arrow. Okay. So what has happened, as you can see on this left hand side, okay, it has captured around 10 odd papers. Okay, but you need to be a little bit concerned about this. That is, it will capture only open source stuff. Okay, the papers or the documents must be available in the open source kind of an environment. Okay, if it is some controlled access is there wherein you require some login ID and password, it will not capture all those sources. So what is important is you need to go through all these, you can say the 10 sources. Okay, so this way you may add as many as 50 odd sources. Okay, right now it is showing 10, but you may add more. Okay, 50 is the limit. Okay, so let's say you are satisfied with all these sources. Okay, now you want to create a notebook out of it. Okay, simply click on this import button. All right, click on import. And then within few seconds, okay, you will find that how many documents it has captured. And my connection is a little bit slow. Okay, that's why it is taking a little bit more time. It all depends upon the traffic at that point of time. Okay. Really sorry, but it is taking a lot of time normally. Maybe the connectivity issue at my end, but by the time I will just explain you the other things, right? नहीं चल रहा। Really sorry, but yes, as I've told you that by the time I will inform you these things, right? So, right, the center panel, you have a chat functionality. Then right hand side, you have a studio functionality, wherein you can do a lot audio in these many Indian languages. Okay. So my internet connection is little bit unstable. Okay. I don't know whether we are connected, but I presume that we are connected. So you may create it using this audio, conversational audio, or on the similar line, you can create a PowerPoint presentation using this slide deck. Okay. Then you may create you can say audio, then video. Okay. But video is by default in English only. Okay. Then you may create a mind map. Okay. Then reports. Okay. So this report part, I'll show you in detail. So it will have some predetermined template, okay, based on the input, or rather the sources, whatever you are using, right? But you can create your own report as well, okay? Whether we are connected or still facing some problem. Hello? Are we connected?
Connected, sir.
Yes. So this you can try, okay? This you can try later on, okay? I'll proceed to some other book because my internet connection is little bit unstable. Okay, so we'll proceed to some other tool as well. Okay, so this is one thing which you may take up as an assignment. Okay, that is you are supposed to build maybe a PowerPoint presentation based on the notebook you have created and upload it on our Samarth LMS. Okay, upload it on our Samarth LMS. Okay. Now it is being created, I think, probably. Okay. So this is the notebook. Okay. Then, as I said, that is you have a chat functionality. Okay. So these are the predefined certain things which you can ask, or you may write your own. Okay. Here somewhere. Okay. Then the right hand side, as I was referring to, that is you may create a research proposal as well. Okay. So how you can do that I'll show you within few seconds. Okay, so you need to go to this create your own. Okay, by default it is in English only, and here you are supposed to type exactly what you intend to create. So you may say create a research proposal. Okay. For MRP. Baba. Use UGC. Hello. He has asked me to inform. तो अभी सर का हो गया क्या? एक सेकंड. Sir, please join. Really sorry, but there are 3 programs simultaneously going on in our center. Okay, so it was basically the resource person's call, so I have to take it. Anyway, so what I was telling you, that is, yeah. So this mind map is generated, then it is generating the report, slide deck, it is underway. Then you can create even quiz and flashcard, and very many things are there. Okay, even this data table is an excellent option. Okay, so you can give a try to this notebook LM. Okay, probably your assignment will be based on this. Okay, Now let us move to some other aspects. Okay, like certain things on the research front. Okay, so wherein illicit, I think somewhere I've mentioned. Okay, so we'll have a look at illicit as well. So for using illicit, you don't require anything other than your Gmail ID or Gmail credential, okay, or any multimedia or social media account. Okay, so it will ask you to log in through your Gmail, or maybe there are many options, or other simply sign up. Okay, so once you create an account, okay, something like this you will find. Okay, then this tool is typically used for analyzing and synthesizing research paper. Okay, so I'll show you the demonstration within a minute or so. Okay, so once again, AI in, let's say, analytical chemistry. Tool has come up with around 10 odd papers. Okay. Internet connectivity is drastically slow. Okay. Anyway, so these are the papers along with their authors, and then other details are there. Okay. Now, as I've told you, that is this, you may use it for analyzing or synthesizing research paper. Okay. Let's say by default, paper title along with the paper authors, those things are there. Then the second column is by default summary. Okay. Now, let's say I want to analyze it in terms of some certain thing extra. Okay. So you go to this right hand side panel. Okay, simply click on edit column, add new, and say research gaps. Okay. So it will now, while crafting this or rather creating this research gap, if you want to pass on some specific instruction, those instruction can be written here. Okay, then what kind of answer type you want, any specified yes, no, maybe. Okay, that can be done quite easily using these. Or if you don't specify anything, it will do it by default. Okay, and then this, now this research gap is the additional column which is being added, and it is populated with the stuff from all those papers. Okay, one after another. Okay. But as far as the limitation of this free version is concerned, okay, you cannot exceed the column limit beyond 4. Okay, right. So if you want to go beyond 4, it will ask you to upgrade. Okay, but there is some solution. Okay, which, let's say you are almost done with this research gap, you may delete this column. Okay, a summary, let's say if you are done, you may delete it and then go on adding the column. So illicit is fantastic for, you can say, analyzing, synthesizing research paper. Okay. Now, I'll take you to few things on, you can say, the agentic front. Okay. Let's say I want to create my own agent. Okay. I want to create my own agent, but now before directly jumping into agent, let's have a look at this Google AI Studio. Okay. So this is what I was referring to. Okay, if you look at this right hand side, now under Google AI Studio, again, using Google AI Studio, you don't require anything other than your Gmail account. Okay, so as you can see here, these are the parameters which we are referring to. Okay, this value of this temperature, right? So you can change it. Okay, yeah. Okay, you can change it as per your need. Okay, by simply dragging it. Okay, so it is max now 2.0, or you can keep it as per your own need. Okay, then the thinking level. Okay, you may keep it as either high, low, or medium, or minimal. Okay, if you make it high, definitely it will take more time. Okay, so these parameters are important. Then this is somewhere I think I have referred to this grounding with Google search. Okay, so all these models, they are basically pre-trained model. Okay, and let's say if you want to take into account the data as on date, okay, then definitely you need to ground it. So that can be done quite easily by using this grounding with Google search. Okay, and then grounding with Google Maps. Okay, these two things are there. Then you may even specify some URL. Okay, and so many things are there, right? So This is all about, you can say, certain parameters, the settings, okay, which we have talked about in our session. Then under models, you have very many models, okay, which you can play with, okay, like say code and chat, maybe image generation. Again, there are a number of models and so on and so forth, right? So you can, so say for example, image creation, you may go ahead with Nano Banana, and there are many things, right? So this is, you can say, Google AI Studio, okay? But in some cases, again, now all these tools are not necessarily free, okay? But let's say if you want to use these tools, another problem with these tools is that is, let's say, if you want to modify your paper, research paper, okay, then definitely you need to upload your stuff. Okay, and when you upload your stuff, you don't know what will happen to it, right? So it is not at all secure, right? So now let's say if you want to use these tools and you don't want to compromise on security or privacy, okay, there is a solution. Okay, that is what RAG basically. I don't know we have that much time, but I'll show you one platform. Okay, what you need to do is for that particular purpose is simply Google, okay, Olama free download, okay, and download the Windows version. Let's say if you are using Windows, okay, download the, now it is not allowing me, but anyway, if you can, right now I'm in office and there are certain restrictions, okay, these are the organizational restrictions. The firewall is not allowing me, but you can definitely give a try. So Olama, go to Olama free download, download it, install it on your machine, but this is a resource hungry tool. Okay, it requires minimum Core i7 processor, 10 generation, and then definitely you have to have minimum 8 or 16 GB of RAM, okay, and sufficient amount of hard disk. That is the mandatory requirement. Now, once you have installed it, okay, I have installed it on my machine. I will show you the demonstration. Okay, show you the demonstration. I hope I'm loud and clear, and we are connected.
Yes, sir.
So the beauty of this Olama is, so if you go to Olama, okay, you will find on the left-hand side corner, you will find somewhere models. Okay, if you click on models, you will find a number of models are there. Now, the beauty of this Olama is it is free and open source. It has been built on FOSS philosophy. So nobody is the owner. Okay, so you can download all those models. Okay, and once you have downloaded it on your machine, okay, you don't require even internet connectivity. No internet connectivity is needed. Okay, once you have installed it. Okay, so Olama framework along with the model. Okay, then whatever interaction you will have, it will be between you and your PC. Okay, whatever prompt you type, whatever responses it will generate, all those things will be there on your local desktop. Okay, on your local desktop. Okay. So this Olama, again, you can give a try. Okay, but I think we are still having 5 to 7 minutes. Okay, so I'll show you certain things on the, you can say, the agentic front. So Zapier is the platform. I don't know whether again my organization whether it allows me to use that. Okay, yeah, fortunately, yeah, it's working. Okay, so Zapier is typically used for you can say creating your own agent. Okay, so for using Zapier, simply go to zapier.com and then simply sign in with your Google account. Okay, sign in with your Google account, and let's say you want to create your own agent, or you want to automate some activity. Okay, and you want to own agent. So if some of you are comfortable with this, again, I may give you some assignment on this as well. Okay, that is create an agent for a specific activity. So simply go to these this new agent tab, click on this new agent. Okay, other way around, you may use this as well. Again, Chrome extension is there. I don't know what has happened to my machine. Okay. So I've clicked on create new agent. Okay. So if you are a first time user, what you need to do is you can use any one of these predefined templates. Okay. Say, let's say I want to automate my some email responses. Okay. So you have a support email agent. So you simply click on that. Okay. And what has happened moment I have clicked on that? Okay. It is showing me the template. Okay. In template, everything has been written in English-like statement. That is when you get a new email. Okay. What is expected from this tool in English only. No programming knowledge is needed. Right. So let's say I want to use this template. Simply click on use this template and simply say create and move further. Okay. Now I may add my own knowledge base. Okay. Let's say I'm running a MMTC center. So my calendar can be added here. Right. So I may automate my, you can say responses to all the participants. Okay. Using this Zapier as an agent, I can very well create. So add knowledge source. You may add up your own, you can say the calendar or the, if you are into a business, some catalogs of your product and so on. Okay. So once you are done with that, okay, simply click on this preview. Right now, I'm not loading anything. Okay, just I wanted to demonstrate the process. Okay, and then simply say test agent. So what has happened? The agent has been created. Now it is, I'm testing it. Okay, I'm allowing access to my Gmail account. So it will go and automatically scan all the emails and it will carry out the activity whatever has been specified in the template. Right. So this Zapier is a fantastic platform for creating agents. Right. Creating agents. Okay. Now the Olama part is left, but I don't know. It is a resource hungry, as I've told you. Right. So now we are almost done. 2 to 3 minutes I have intentionally kept for the question answer. Right. If you have any specific query, you can fire it now. So let me sum up. Okay, we have started with some basic tools like say Notebook LM, then we move to you can say Elicit, and then later on we have seen Zapier as well. Okay, Olama you can give a try. Okay, Olama you can give a try. Olama is fantastic. Using Olama, you can build your own large language model. Okay, the RAG concept, whatever I have demonstrated. Actually, I'm really sorry that I am not in a position to show you right now, but because Olama, I am having some problem. But as a matter of fact, it is there on my machine. Let me give a second try. If it works, I will show you. Okay, by that time, if you have any query, you can fire it now.
Sir, good
Hello,
evening.
sir.
Hello.
Yeah, good evening. Please.
Sir,
Yes, ma'am. Please.
sir,
Please go ahead.
I want to ask which will be the best tool for creating a research proposal, sir? You told a Notebook LM?
Notebook LM can be used, madam, for that purpose, but I will not recommend Notebook LM for that activity. I wanted to demonstrate that is how you can create your own customized report. And that's why I have demonstrated it. But there are many tools. Okay, one should not see Notebook LM is still under development, and it is available free of cost. Right.
Okay.
So even if you look at, yeah, Olama is now ready. Okay, this is how the Olama will look like now. Okay, now Olama actually we have tried. Okay, so this is the structure of a Olama. Okay, and as you can see here, I have downloaded only one model here. Let me show you. Okay, but Olama can be used for creating your own LLM, large language model, your own. Okay, using some pre-existing model. Okay, so but it is very resource hungry. Don't try it on mobile or anything like that, right? Even if you are having i3 processor, don't try it. Okay, minimum i7 as I'm telling you repeatedly. Okay, so those model basically will appear here. Okay, unfortunately, it is taking long time. Yeah, there we are. Okay, so this Llama 3.2 I have installed. Okay, as you can see here, Llama 3.2. Okay, so now whatever interaction I will have, okay, even if you look at the chat history there on the left hand side, okay, it is between me. Now here onwards, I don't require any internet connectivity. Okay, because the framework I have downloaded, the model I have downloaded, and this is the model which I have created, what they call it as the structured LLM, right? This is the another model. Okay, so this has been built on 5.3. Okay, so you need to download. There are few steps, but within 10 to 15 minutes, again, you can build your own large language model. So you can train it with your own data or national data or your personal data, and you may use it. So the beauty of this Olama is you are safe and secure. Okay. So there is no threat to your privacy and secrecy. Now here you can upload your paper because everything is within your control. Okay, paper you upload or the conversation you have. Let's say you are generating a question paper. So everything is confidential. It is between you and the tool only, right? So I'll not cross my limit. Okay, the time allotted to me was 1600. Okay, and we are dot 1602. So if there are any specific query, I'll be happy to answer. Okay. So this is all from my end.
Good evening, sir.
Yes, sir.
Good evening, sir. Sir, the information which
Please go
we
ahead, sir.
share with AI tools,
Yes.
the information which we share with the AI tools, is that information secured or not? That chat has done with the AI tools.
Sorry.
Generally, we chat with the AI tools. So whether
Yes, sir.
that chatting is secured with that AI tool or not?
It is not at all secure, and that's why I'm saying कि if you carry out, it is not at all secure, sir. It is vulnerable in terms of security. है ना? तो आपको सिक्योरिटी के लिए ही हमने आपको ओलामा बताया है ना? ओलामा को आप डाउनलोड करिए, ओलामा में आपके मॉडल्स डालिए, फ्री वाले मॉडल्स, है ना? और उसका प्रयोग करिए, ठीक है? लेकिन चैट जीपीटी पे आपका कोई पेपर, रिसर्च पेपर मत डालिए. यू विल फेस प्रॉब्लम, लॉट ऑफ प्रॉब्लम, राइट? In terms of security and privacy. Okay.
Okay. Okay.
So you will get some assignment. Okay. You may take one assignment as create a notebook LM of your choice. Okay. And then maybe prepare the PowerPoint presentation, and I'll give you the exact stuff on the group. Okay. So that you can accordingly upload it on the portal. So thank you very much. And I hope
Hello,
you have
sir.
enjoyed this session. Yes, sir. Yes, sir.
Sir, suppose I download any AI tool in my laptop or mobile. Can my data I can save or not?
As long as you are not online, it is safe. Moment you say, for example, Olama, there are two versions. One is the cloud version and the second is the desktop version. If you are using desktop, you are safe. Moment you are using the online data, online or the cloud version, you are not safe. Okay, it is vulnerable.
Okay. Okay.
Okay, sir. I hope I have answered your queries. So thank you very much.
Okay, sir.
Now probably, yeah, yes, sir. Bola.
Thank you, sir. Good evening to everyone. As we wrap up this fantastic third session of second day of our refresher course, I am Mr. Amar Pawar, here to offer a formal yet very heartful thank you. First and foremost, a huge thank you to our resource person, Professor Dr. Mohammad Atik sir. Looking at such background with over 34 years of teaching experience, multiple degrees in engineering, management and law, and over dozens of patents, it is safe to say that we were in the presence of true academic powerhouse today. Beyond this impressive resume, what we really stood out that today was the sheer passion of sir, clarity that sir has brought in the session, and deep diving into the fields like artificial intelligence, generative AI, different tools that we can use as AI models. At the same time, we all know that sir has busy schedule. Still, sir has managed his time for us and mentored us, guide us today. So once again, I'm very thankful to sir to giving a warm and great knowledge to all of us in the field of artificial intelligence. I also want to thank our organizers of this refresher course, UGC, MMTC, Sant Gadge Baba Amravati University, for keeping everything so smoothly. And also I am thankful to Amita Joshi madam for wonderful introduction of this session. Finally, I am thankful to my fellow participants. Once again, thank you, Professor Dr. Atik sir, for wonderful session. We have taken a lot of things from your session, sir. Thank you, sir.
Thank you. Thank you, sir. Just allow me to leave. Thank you. Thank you, one and all.
Thank you so much, sir.
Thank you so much,
sir.
Thank you.
Thank you so much, sir.
Thank you, sir. Thank you.
Thank you, sir.
Thank you.
Hello, Zugade, sir.
Yes. Good afternoon, madam.
Sir, namaste, sir. Good afternoon. So shall we start with the session, sir?
Yes, if participants are ready, I am ready.
Yes, sir.
Yes.
May I now request who is going to introduce, sir, please?
Dr. Vinil Kaushal, ma'am. May I audible, ma'am?
Yes, you're audible. Please go ahead.
It is my great pleasure and privilege to introduce today's special resource person, Professor Dr. Manohar Jugade, sir, Professor of Analytical Chemistry in the Department of Chemistry, ITM, Nagpur University. Professor Jugade is highly academician and researcher with outstanding contribution in the field of analytical chemistry. He was awarded his PhD by Nagpur University in 2004. With an impressive research career spanning over 2 decades, Professor Jugade has successfully completed 5 major research projects and currently serving as the principal investigator of the DSC pursuing project and co-investigator of an ANRF pairs projects. His academician achievements are truly remarkable. He has published 126 research papers, authored 70 books, guided 11 PhD scholars, and 1 postdoctoral research scholar. Professor Jugade has received several prestigious recognitions, including the Best Research Award 2023-24 by the RTM Nagpur University, the Best PG Teacher Award 2023-24 by the All Indian Association of the Chemistry, and Honor Fellowship with the Indian Society of the Analytical Chemistry. Apart from his academician excellency, he has made significant contribution to university administration. Professor Jugade is an effective lifetime member of several prestigious scientific organizations, including Indian Science Congress Association, Indian Council of Chemists, Indian Association of the Chemistry Teachers, etc. Today, we are indeed fortunate to have such an eminent scientist, teacher, and inspiring academician with as a resource person for this refresher course. On behalf of organizing committee and all participants, I extend warm and heartfully welcome to Professor Dr. Manohar Jugade, sir. You are welcome, sir.
Thank you very much, sir, for the nice introduction. Can I start my lecture, madam?
Yes, sir. You're welcome, sir, and thank you very much for giving your precious time, sir. Please, sir.
Okay. Is my screen visible to all?
Yes, sir, it's visible.
Yes, sir.
Yes, okay. Sir. Yeah.
Yes, sir.
Yeah. Thank you very much. Thank you. So it's almost 4:15 in the evening, and I can understand it's very difficult for the teacher fraternity people to listen 4 lectures of 1.5 hours a day, and it's very difficult at the evening time especially. And I was just joining the lecture of Dr. Arif, who was presenting excellent work on the field of the AI and AI tools, the hot dog. And after those wonderful lectures, a person talking about statistical analysis, so it's very boring, and I will definitely try to make it as halka fulka as possible because it's an evening session. So I will definitely suggest all the participants not to get panic, just listen it carefully, and you will find at the end of the lecture that you have got something regarding the core analytical chemistry component. So the topic is statistical analysis for chemists. So it's basically, as the refresher course is for chemistry people, I will just restrict my topic to the application of the statistical analysis in the core chemistry and chemistry research. Some of the topics are related to, or you have already learned in MSc classes, and some of you might be teaching some of the topics. Just I will try to go into depth of that. So at any moment, if you have any queries, you can definitely ask the queries by unmuting yourself, because since I am sharing my screen, I will not be able to see your faces, but definitely at any moment, if you have any doubt, you can just unmute yourself and ask for any query. So let us start with this topic. Just go through this slide once. I will give you a minute to read it. Okay, so let us see what this fellow is asking. Is he is saying that don't move, I will fill you full of 98% lead and 1% antimony and silver, vagara vagara. And this fellow is standing in front of him, must be an analytical chemist. So he is asking, wait a minute, are those values certified? So that is actually the role of the analytical chemist. So whatever values you put in, are those values certified, or are those values really reliable or reproducible? That is actually the role of analytical chemist. So when I say any chemical analysis, anything which comes to me as an analytical chemist, I call it as a sample. Now this sample can be anything. It can be a bottle of water, it can be a solution like this, it can be a blood sample, a few ml or a few drops, it can be as large as an complete ocean. So, or it can be a dead body, it can be a body part. So anything which comes to me will be called as a sample. And this sample has a typical composition of two components. One is a component which I want to analyze. That component is called as the analyte. And whatever are the remaining components in the sample, that will be called as a matrix. So my role as an analytical chemist is to determine analyte in the presence of this matrix in the sample. So there are two questions associated with any sample. What is present in the sample and how much is present in the sample? So both the questions are asked, and they are associated with two types of analysis called as the qualitative analysis and obviously the quantitative analysis. But whenever any sample comes to me, the very first question comes into my mind, okay, what is the technique which is available for analysis of this sample, whether qualitative or quantitative? In any case, what are the techniques available? For example, if a water sample comes to me and a question is that how much is the concentration of iron present in this water sample? So that is a simple question. Some lake water sample coming to me and I want to determine how much of iron is present in that sample. The very first question comes into my mind, what are the techniques which are available for estimation of iron in that sample? So for that, I definitely go to the best analytical chemistry book that we call as the Vogel's textbook of quantitative chemical analysis. So when I use that Vogel's textbook, so definitely Vogel's textbook is the textbook which is called as, it is actually not the textbook, even if the name is Vogel's textbook of quantitative chemical analysis, but it is actually a reference book. And the difference between a textbook and reference book is that the textbook you read from first page to last page, but the reference book you read from last page to first page. Actually, you go to the index and see what are the techniques which are available for estimation of iron in water samples. So in Vogel, I get a large number of techniques by which you can estimate iron in water sample. So the question is not availability of the techniques. There are multiple techniques available. The question is selection of the technique is an important property or important criteria. So which method should I select? So that selection of analytical technique is the major problem of an analytical chemist in the beginning. So the technique selection is based on certain criteria. These criteria are what is the accuracy required, what is called as the agreement between the observed value and true value, what is the required precision, what is the detection limit of the technique which is available, what is the sensitivity of the technique which is available, and of course, if you are working in a college or in any laboratory, definitely the cost which is involved in the technique or the instrumentation procurement or the requirements, that is another aspect definitely important for the selection of analytical technique. So based on all these aspects, we have to select a proper technique among a large number of available methods in the textbook, or say you can say the literature which is available with us. So now these analytical techniques we classify into two types. One are called as the classical methods and second are called as the definitely the instrumental methods of analysis. Classical are the old methods which are based on mainly the chemical properties of the analyte present in the sample. So when I say analyte, it is the species which I want to analyze. For example, in the previous example, I have said iron present in water. So water is a sample. Analyte is definitely iron. So estimation of iron. So how to estimate this? So in chemical properties, based on the chemical properties, we call as the classical methods of analysis. And these classical methods can be further classified as volumetry and gravimetry, the basic fundamental analytical chemistry. And the instrumental methods of analysis, that is they are based on the physical properties of the analyte. So they can be further classified as optical methods, chromatographic methods, radiochemical methods, and electrochemical methods. So this is the general way of classifying all those analytical techniques. So when I compare these classical methods with these instrumental methods, the most important aspect of them are sensitivity and the detection limits of these two. Take an example. If I come up with, say, two test tubes, I give you two test tubes. One is containing, say, 1 molar copper sulfate solution, and second is containing, say, 2 molar copper sulfate solution. And I ask you, without carrying out any analysis, can you determine which is 1 molar and which is 2 molar? Anybody can answer this. I am going to make it a communication. Yes.
Whether on color.
By color.
Definitely, both of them are colored, and copper sulfate is a colored solution. So by just visualizing the optical density, or what we call as the intensity of the color, you can say that this is 1 molar and this is suppose 2 molar. Definitely you can tell. Then again, I replace this 2 molar with another test tube. Instead of 2 molar, now I have 1 molar and 1.1 molar. Now can we distinguish it now?
By titration, sir?
No, without carrying out any analysis.
No, it's very difficult, sir.
Yeah, maybe difficult, maybe we are able to, may not be, है ना? Probably आपने बहुत अच्छे से उसको देख के you determine कि हाँ ये थोड़ा सा dark दिख रहा है, so it should be 1.1. Again, I come up with two test tubes, 1 molar and 1.01 molar. Now it is almost impossible to determine which is 1 molar and which is 1.01 molar because the concentrations are very close to each other. Now in that case, the way of analyzing which is 1 molar, which is 1.1 molar, so that is actually the limitation of our eye. So now I want to analyze them. So what is the method? Replace the eye with an instrument, right? So if you replace the eye with an instrument, the technique becomes instrumental method of analysis, and you can use some colorimeter or spectrophotometer to distinguish between 1 molar and 1.01 molar. So by using an instrument, you can go up to a particular difference, which is even, it can be even closer than this. So you can definitely distinguish between 1.1 molar and 1.001 molar also by using some instrument. So what is the basic difference between our eye and the instrument is the difference in the sensitivity of that. Now, how will you define the sensitivity? These two things are always, they go hand in hand, but they are always misinterpreted. One is called as the detection limit and second is called as the sensitivity. So when I say detection limit, it is the minimum detectable concentration. But when I say sensitivity, is the ability to distinguish between two close concentrations. So these two things are different in case of classical methods of analysis and instrumental methods of analysis. So when you go from classical to instrumental, we improve the detection limit. So when I say improved detection limit, means you are going at lower concentrations, and the improved sensitivity is the ability to distinguish between two close concentrations. I think I am very clear over here. So let us try to understand the basic concept of what is called as the errors in chemical analysis. The reason of learning errors in chemical analysis is how to overcome those errors or how to avoid those errors. I will just go a bit fast initially because these are the basic concepts and everybody knows about it. Just I will go through it very rapidly. So what we call as the error is the numerical difference between the observed value and a true value. So what is the observed value is the observation which you get through experiments, and true value is the actual concentration of the analyte present in the sample. So this true value is most of the times not known to us if the sample is unknown. If we prepare a standard sample, then we know what is the actual concentration. But if we take unknown sample from outside, definitely we do not know this true value. And if we know the true value, then we are able to determine how much of error is involved in our analysis. So these errors may have reasons or may not have reasons. If the errors have reasons, they are called as systematic errors. And if they do not have any reasons or known reasons, then they are called as the random errors. So these errors can be further classified depending on the reasons as personal errors, methodic errors, instrumental and reagent errors, and operational errors. So these errors, if we know the cause of the errors, they are called as the systematic errors. We will just go through the systematic errors. So if the cause is known, then they are called as the systematic errors. So if the cause is known, it means that they can be avoided. So it is possible to avoid these systematic errors. So they are also called as the determinate type of errors. So these determinate errors are generally constant, and all the observations made under the similar conditions, they are further divided as into different types. The first type of that is very common error by the person called as the personal error. And this personal error is because of inability of the analyst to carry out or to take the observations accurately. And a simple example is, if you are not able to judge the color change at the end point very properly, then definitely there will be overrunning of the end point and you will get some error, and this is called as the personal error. So this is quite obvious. The students definitely come to us many times, sir, ye color change ho gaya hai kya? So the person is continuously looking at the color, must be knowing what is the color change. So he should not come to us, but he should be able to judge the color change. Many times you must have seen that the students adding an extra drop so that the color becomes, take an example like say oxalic acid titrated with NaOH. The most common titration starting from standard 11, 12th up to BSc and MSc levels. This is a common titration. And in that titration, we will find that the phenolphthalein is used as an indicator, and an extra drop of NaOH will impart a dark pink color. No, there is always a faint pink color is expected. And at the end point, if we add one extra drop, so that will lead to an error that will be called as a personal error, and this value will vary from person to person. So the question is how to overcome this error. Can anybody suggest? How to overcome personal error?
So by taking concordant readings.
Personal error cannot be avoided
to remain very cautious during experiment. Sir, to remain very cautious during experiment.
Yeah, the best way of overcoming these errors are practicing multiple times. It's the only way of doing or overcoming personal error is practicing. Because see, when you carry out an iteration once, you will not be able to identify. If you carry out iteration 5 or 10 times, you will find after a few number of iterations, the results which, or you get some correctness in your observations, and the best way of doing is either replace the person or train the person. So the best way is training. Another error is the methodic type of error. This error is involved when the correct method is not followed. And now the problem is not in the person, but the problem is in the method itself. And mainly in the gravimetric analysis, you will find this type of problems like co-precipitation, post-precipitation. These are the phenomena like solubility of the precipitate. Now the thing is not known to us, but if a co-precipitation is taking place, the co-precipitation is a very interesting phenomena. I like it like anything. This co-precipitation is an interesting phenomena. What is this co-precipitation? If you have a sample containing suppose A, and if you add a reagent B does not react, or B reacts with A, or B does not react with A, forming a compound suppose BA. But this BA is completely soluble in water, so it does not precipitate out. But if the solution contains some C, and this B, when added to a mixture of A and C both, then this C forms a precipitate BC. So C is getting precipitated. Now since C is getting precipitated, A also start getting precipitated in the same solution. Now if C is not there, A does not undergo precipitation. But if C is present, A undergoes precipitation. This phenomena is called as a co-precipitation phenomena. This is interesting. This is actually what we call, it is the say, you can say neighboring group participation is there. You can say in organic chemistry we say it is a neighboring group participation. So this co-precipitation phenomena is just like neighboring group participation, and it takes place simultaneously during the main precipitation. Similarly, post-precipitation is after the precipitation of main precipitate. Again, it is the precipitation of that compound which is normally not soluble, not sparingly soluble salt, which generally does not undergo precipitation, but undergoes precipitation in the presence of main precipitate. So this is called as the post-precipitation. So these things are not known to us, and as a result, the fault is not of the person, but fault is involved in the method itself. In gravimetric analysis, because of these phenomena. In titrimetric analysis, it is because of the side reactions and slow rate of reaction. This is another aspect. If the rate of reaction is slow, we cannot carry it out in a titrimetric manner, or you cannot determine the concentration in a titrimetric method. Quite obvious, because when you add the next drop, before addition of next drop, the first drop must have neutralized. But if the rate of reaction is slow, then the neutralization is incomplete, and you will have to add extra drops to complete the process. Third type of errors are because of the uncalibrated instruments and the impure reagents. So it is not that the instrument should be exactly accurate, but it should be calibrated. Similarly, reagents may not be pure, but the purity must be known to us. That is important. And last is the operational error. Large number of errors involved in operations during mainly the classical methods involve these type of errors, like loss of materials during various steps of analysis, mainly in gravimetry, errors in burette reading, weighing of crucible in hot condition. We know that when we weigh a crucible in hot condition, the weight is it decreases because of the buoyancy force. Improper washing of the precipitate and errors in calculation. All these come under operational errors. So these are the errors whose cause are known, and so they can be avoided. There can be another classification like additive and proportional errors. So if I say what is called as an additive error is a constant error added in all observations irrespective of how much is the analyte. And what is a proportional error is the error which goes on increasing with quantity of the analyte. For example, if I weigh 1 gram, if there is an error of 0.1 gram, and if in 10 gram also the error is 0.1 gram, then such error is called as an additive error. It can be on positive side, it can be on negative side. But proportional error, the error goes on increasing. For example, in 1 gram, there is an error of suppose 0.1 gram. So in 10 gram, the error will be of 1 gram. So this is called as a proportional error. So the error goes on increasing with quantity of the analyte. Now, if the cause is not known, even after that, some errors are observed and they are called as the random errors. These random errors, the cause is difficult to know. If you see these random errors, So if you see this zero error is accuracy. So you will find that these random errors distribute or follow what is called as the Gaussian distribution. This Gaussian distribution is on both sides, the probability of getting error is equal. Suppose the endpoint or the actual endpoint of a titration is 10.5. So sometimes we can get some 10.4, you can get some 10.6, sometimes you will get. So if you have a batch of 50 students, everybody is carrying out the same titration with same instrument, he will come up with readings which are very close to this, or some of the values slightly away from this. So you will find that these errors have some important properties. Like the small errors are more frequent as compared to larger errors. If you see that the small error, so error is small, then its frequency is higher. If the error positive or negative, error is small, frequency is higher. But if the error is a large, frequency is less, either positive or negative. Large error is less frequent, small error are more frequent. Quite obvious, 10.5 का 10.4, 10.6 आने की probability सबसे ज्यादा रहेगी, ना कि 11 और 12 आने की probability. Similarly, positive and negative errors are equally likely to occur. Random errors की speciality है, they lie on both sides of the true value. Thirdly, narrow sharp peak indicates higher precision. So you can have a peak like this or a peak like this, a sharp narrow peak or a broad flat peak. So if the peak is like broad and flat, it leads to what is called as a low precision. And if you have a sharp peak, it is a high precision. Precision is reproducibility. We will discuss it in details. Now the question is, how will you overcome these random errors? Because the reason is not known to us. So how to overcome these errors? Can anybody?
Sir, can we change sample size?
By changing the sample size, the random errors cannot be avoided, because the cause is not known to us. See, it can vary. You can decrease it by taking a larger sample volumes. The proportional error, or what we call as the relative error, can be reduced, but you cannot avoid them. Any other option?
Sir, I think it is following Maxwell-Boltzmann curve. The errors are following like Maxwell-Boltzmann distribution curve.
No, these random errors have a special probability. Random errors always follow the Gaussian distribution. That's why you can overcome them. There is only one method by which you can overcome random errors.
But if errors are minimized, then this peak will become more sharp.
Yes, that is okay. If you minimize it, you take large number of observations. But how to reach to this value? How to reach to this value?
By repeating experiments, by repeating.
Exactly. The only way of overcoming random errors is to take a large number of observations and average them, because you can see that the positive and negative errors are equally probable in case of random errors. So if you take average of large number of observations, you are definitely going to reach to this value, or to very close to this value, you can say. It's not possible to get exactly true value, but what you can get is to very close to true value by averaging a large number of observations, provided there are no systematic errors involved. You will have to avoid all the systematic errors. Then only errors which are involved are the random errors. And if only random errors are there, they can be nullified, because positive and negative random errors are equally probable. I think I am very clear now. Yeah. Another important concept in this topic is the accuracy and precision. So it is accuracy. This is again an important concept of analytical chemistry. The agreement between the observed value and the true value. which is generally not known to us, is the accuracy, and agreement between a large number of observed values is called as precision. So accuracy, when you are accurate, you are reliable. And when you are precise, the results are said to be reproducible in nature, reproducibility. And accuracy is expressed in terms of error, while the precision is expressed in terms of standard deviation. In other words, you can say that lower errors means more accuracy, and lower standard deviation means more precision. This is important aspect. Determinate error always affects accuracy. Determinate means the systematic errors. The cause is known, then accuracy is affected. But determinate error may not affect precision if it is constant for all the observations. Let us try to understand this concept. This is very important. Determinate errors always affect the accuracy. If, take an example, determinate error या जिसको systematic error हमने बोला है, systematic error में if a person is not able to determine the end point of a titration correctly, तो क्या होगा? End point, suppose exact end point is 10.5, but he is not able to judge this end point. So what he will get? He will come up with some results like 10.6 or 10.7. So what has happened? The true value is 10.5 and he is coming with a value 10.6. So accuracy is affected, definitely. So determinate error has affected the accuracy. Come to the precision. Now, if the person carries out the titration, suppose 10 times, he will definitely get 10.6, 10.7, 10.6, 10.7, 10.6, 10.7, this type of values again and again. So if you see the observations or agreement amongst themselves, you will find that the results are not accurate, but they are precise. So they may not affect the precision. Determinate error may not affect the precision. This is an example. You will find this type of diagram in many of the textbooks or the reference books. There is no correlation between accuracy and precision. I take an example. If I have a bullet and I have to hit this bullseye, and I hit here, 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 here. So you will find that I am not accurate because my value is not reaching to this, or my bullet is not reaching the desired value. And I am not precise because these values are not matching with each other also. So the example of this is, first, if I carry out a titration, say 5 times, suppose true value is 10.4 and I am getting the values as 10.5, 11.3, 10.9, say 11.5, 11.9, and the mean value is 11.2, which is much away from this true value. The standard deviation is very high. So what I can say is, this first analyst is neither accurate nor precise. Accuracy भी नहीं है, precision भी नहीं है. It is exactly similar to what is presented over here. In second case, you will find that these values are away from the desired or true value, but they are matching with each other. They are in agreement with each other. So you can say precision is there, but accuracy is not there. So you will see this analyst number 2. The values are 11.0, 11.1, 11.3, 10.9, 11.2. The values are close to each other. But if you see the mean value is 11.1, which is away from the true value. So it means that he is not accurate but precise because the standard deviation is very low. Third case, you will find these are the 4 values. Now, if you see that they are not agreeing with each other, but if you take the mean of all the 4, it comes over here, which is matching the true value. So let's take an example. Say 10.0, 11.1, 10.4, 10.6, 10.9. So if these are the 5 observations, you will find that they are not agreeing with each other. But if you take the mean of all those 5 values, you will find that it is 10.4, which is matching with the true value. So you are reaching the accuracy, but precision is low because standard deviation is very high. Coming to the fourth person, hitting all the bullets like this, so agreeing with each other, also very close to the true value. So it means that the analyst is getting like 10.5, 10.4, 10.4, 10.5, 10.3. The mean is 10.4, matching with the true value. And also the standard deviation is very low. The values are agreeing with each other. So it is accurate as well as precise. Now, my question is, if you see this, when I say accuracy and precision, what I am actually doing? If I am taking a large number of observations, I will get observations like this. Definitely the Gaussian distribution curve. And when I am talking about accuracy, I am comparing mean of my all observations with the true value, or what you call as a reference value or certified value. So this is accuracy. But when I am talking about precision, I am just trying to compare all the observed values taken under identical condition, and it has nothing to do with the true value at all. This is fundamental difference between the accuracy and precision. My question is, as an analytical chemist, what will you prefer? If there is a method which is accurate or a method which is precise?
Sir, I will prefer precise method.
Sir, I will prefer accurate method.
Okay, so there can be both the things. No, some of you may prefer accuracy, some of you may prefer precision. Any other answer? A good analytical chemist or a good analytical technique should be neither only accurate nor only precise, but it should be both. So there is no compromise. Ye chalega, wo chalega is not allowed in analytical chemistry at all. Now, how to reach this?
Method should be precise.
Pardon?
Our experiments should be precise.
Yeah, I am talking about a method. The method which is giving precise results, a method which is giving accurate results. Okay, so definitely it has to be both precise as well as accurate. Let us see what do I mean by this. When I say it has to be both precise and accurate, it means that, see, this is your true value and this is your observed value. So when I say accurate, it means that this observed value should be extremely close to the true value. And when I say precise, it means that my distribution should be compact. So the values should be very close to each other. That is what is meant by accuracy and precision. So my method should be both accurate as well as precise. Another important concept and much less understood concept. Significant figures. Just 1 minute, go through this. Read this at the bottom. एक न्यूज आई है एंड द न्यूज शोज दैट न्यूज एंकर जो है वो बता रहा है कि एस्टिमेट सजेस्ट some 4,28,620 people have perished, या मर गए किसी जगह पे, है ना, they died. So the word is estimates suggest this number. Now when you say estimate, the number cannot be so precise. नॉर्मल पर्सन की रिएक्शन है, ओ माय गॉड सो हॉरिबल. हमारा एनालिटिकल केमिस्ट है, स्टैटिस्टिशियन है, what he says, oh my god, what, there is no way they know that many significant digits. This is interesting. If he says about 4 lakh people, about 4.2 lakh people, that is okay. But what is a statistician is thinking. So the way of thinking is entirely different in a normal person and a statistical person. Let us see what is this significant figures means. Significant figures are the number of digits necessary to express the results of the measurements to the precision with which it has been made. Or some scientists define significant figures as all the digits which are certain and one digit which involves some uncertainty. Sorry, एक word यहाँ पे miss हुआ है. Okay, so this is uncertainty word. So all the certain digits and one uncertain digit, that is called as significant figures. Now, if you see this definition, it is very difficult to understand. The number of digits necessary to express the results of the measurement to the precision with which it has been made. It's interesting. Take an example. Normally, a common person अपन regularly एक titration करते हैं. Suppose oxalic acid NaOH का titration हमने किया, and 4 students A, B, C, and D, using the pipette and burette and a titration of oxalic acid with NaOH, came up with these 4 observation tables. Just go through these tables and let me know who has reported the values correctly.
Student number A.
Student A, sir.
A. So let us make numbers. Just a minute.
I think 11.
Student number D.
चलो ए वाले कितने लोग हैं? ए का मुझे 2 लोगों ने बोला है.
सर
A.
B. Good.
Sir.
D, sir. D.
D.
D, sir.
D.
Sir, D.
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 I think A or B. A and B.
Sir,
Sir,
D.
D should be correct. D.
D.
D, sir.
D.
Sir, D is correct.
D is correct. Okay.
D
Nobody.
is right.
हाँ A. चलो A और एक बार.
Sir, D.
D.
Sir, D, sir.
D.
A is correct, sir. A.
A.
D, sir. D.
D
Sir,
correct.
A is correct.
A is correct.
A is correct. चलो A is correct. A is correct. A को बहुत ज्यादा majority आ रही है. हाँ. So there is competition between A and D now.
Sir,
A is correct.
Yes, sir. A is correct.
Sir, make that
A is
bundle
correct.
of 5 by slashing, so we can count easily.
Yeah, no, I am I just want to see not quantitatively but qualitatively where is it going.
Just for fun.
According to least count of burette, A is correct, sir.
A is correct. Okay.
A, sir. A.
A.
Yes, sir.
A.
No, sir. D.
D. So let us see. Okay. अच्छा चारों करेक्ट वाला कोई नहीं है क्या?
No, sir. A is correct.
So, sir, according to this image,
हाँ. Yes.
Least count of burette is up to 1 decimal point. That's why A is correct.
A is correct. Okay. चलो. All the 4 are wrong.
सर वो 10 ml pipette है.
What is correct is this.
Sir, how?
How? The question is, see, if you see the pipette, that's why I have shown this burette and pipette here. Now, in pipette, there are no markings of 0.1 ml. 0.1 के कोई markings है क्या यहाँ पर? And if there are no markings of 0.1 ml, you cannot report it up to first decimal place, क्योंकि फर्स्ट डेसिमल का आपके पास ऑब्जर्वेशन ही नहीं है. अब आते हैं हम हमारे डेफिनेशन पे. क्या डेफिनेशन है? The number of digits necessary to express the results of the measurement to the precision with which it is made. जिस प्रिसीजन से ऑब्जर्वेशन लिया गया है उस प्रिसीजन से एक्सप्रेस करना है हमें. सो हियर इट हैज टू बी ओनली 10, क्योंकि आपके पास एक ही मार्किंग है यहां. बट इन केस ऑफ बिरेट यू आर हैविंग फर्स्ट डेसिमल के रीडिंग्स है, सो फर्स्ट डेसिमल हैज टू बी रिपोर्टेड इवन इफ इट इज 0.1, sorry, even if it is 0. So 11.0 has to be mentioned. That's why you will find this B में ये बराबर है. 10 10 10 is correct, but 11 11 is wrong because you have to report up to first decimal. In other words, थोड़ा सा सिंप्लिसिटी में अगर हम जाएंगे तो इंस्ट्रूमेंट ने जो रीडिंग हमको दिया वो पूरा का पूरा एक्सप्रेस करना है मुझे. उसके डेसिमल 0 हो, नॉन 0 हो, कुछ भी हो. इफ आई डिसॉल्व 0.63 gram of oxalic acid by using a 4 decimal balance, even if the last two digits are 0.6300, you have to report it. By looking at these values, I will be able to tell that this observation has been taken by using an instrument of 4 decimal. तो मेरा ऑब्जर्वेशन देख के मुझे पता चलना चाहिए कि मेरा इंस्ट्रूमेंट कौन सा यूज हुआ है, या इंस्ट्रूमेंट से मुझे पता होना चाहिए कि मैंने ऑब्जर्वेशन कहां तक लिखना है. दैट इज कॉल्ड एज द सिग्निफिकेंट फिगर्स. इज इट क्लियर टू ऑल?
यस सर, यस सर.
Yes, sir. Yes, sir.
आगे बढ़े. चलो लेट अस सी. सो रूल्स ऑफ सिग्निफिकेंट फिगर्स हमें पता है कि इन बिटवीन जो भी जीरोस होते हैं, मेनली जीरोस के बारे में रहता है. ऑल द नॉन जीरो डिजिट्स आर सिग्निफिकेंट. इंटरमीडिएट जीरो या इन बिटवीन मिडल जीरोस दे आर सिग्निफिकेंट. लेफ्ट हैंड जीरोस आर नॉट सिग्निफिकेंट. इट्स वेरी इंपॉर्टेंट. राइट हैंड जीरोस आर सिग्निफिकेंट if the number contains a decimal point, and the right hand zeros or trailing zeros may or may not be significant if the number does not contain a decimal point. This is interesting, huh? Let us see once again. Middle zeros are significant, no problem. Left hand zeros are not significant. Right hand zeros are significant if the number contains a decimal point. Conditional hai. And right hand zeros may or may not be significant if the number does not contain a decimal point. We will try to understand this concept by taking a few examples. Let us see how many significant figures are there in this number. 6. Yes.
4.
Definitely this because they are
4.
Yeah. So definitely 4 significant figures. No problem. यहाँ कोई problem ही नहीं है. What about this?
2.
2. Yeah.
2,
Only
sir.
2. Yes. The reason for this 2 is the same value I can put as 2.3 into 10 raised to power minus 3. ऐसा लिख सकते हो क्योंकि when I write it like this, say for example, यहां पर अगर आप देखेंगे कि in this particular case, when I say significant figures are 4, as soon as I say 4, it means that the 4th digit is uncertain. These 3 are certain. If I say this is certain. Sorry, this is uncertain, last digit. This is certain. and this should be certain. But if I put it like this, 2.3 into 10 raised to power minus 3, this is uncertain and this is certain. The same value can be represented with one certain and one uncertain digit. So only two digits, one uncertain, one certain. So two significant figures. What about this?
Sir, takes
3.6.
4.
6, sir.
Any number.
6.
4.
6.
6.
6. 6.
See,
6,
if
sir.
as soon as I have reported is it as 33.4500, it means that I have taken observation by using an instrument which can report up to 4 decimal places. And it means that if it is reporting